So I know there's still a few to join. But good morning, everyone. I'm Denise Beaton, the Crop Protection Specialist with the Ontario Ministry of Agriculture, Food and Rural Affairs, OMAFRA. So hope everyone's having a great day so far and you were able to join this Zoom meeting with no issues. And hopefully you were able to access the handouts that I provided using that Adobe link in the email. I also did send a revised farm visit biosecurity protocol this morning and the agenda. So please refer to the revised protocol and yeah. So that's about it. So let's get started. So in this workshop, we cover various topics such as the importance of integrated pest management, so IPM, so you can understand why scouting is so important. We're also going to be covering diseases, insects, weeds, soil diagnostics and sampling, nutrient deficiencies and tissue sampling, farm visit biosecurity, pesticide safety. I think that's it. So this is the introductory course that covers the basic information that you need to know as a scout. We do offer other OMAFRA scout workshops that are tailored to the specific crop. So if you're interested in joining any of those, if you haven't already done so, you can register for those on the vegetable blog and on fruit blog. So I did provide those links in the handouts that you should have received. I hope everyone had a chance to listen to the couple videos I referred to in the email, or you got to look at the PDFs of the talk, since we will be reviewing some of this content and doing a Q&A session with time for discussion. And just a few housekeeping things. Please don't share your screen unless you really want to. No, don't do that. And this session is being recorded, so you can turn off your camera if you wish. And this may help if you tend to have internet connection issues as well. We're also going to be running some polls, so you're going to be asked questions. The polls are anonymous, so don't worry if you get a wrong answer. But after you've answered, the box tends to stay there, so just make sure you close it out so it's not blocking your screen. We do have everyone muted, but if you do have questions, you can submit those in the chat, and we'll monitor that, and the presenters will address that at the end of their presentation. And you should have got the agenda, and so I'm just, I forgot to share my screen. Okay. Okay, so you should have got the agenda with the email this morning, and it was also at the Adobe link that I provided. So we will have a 10-minute break at 11 and also a 30-minute lunch hour. So just to give you time to grab something to eat and walk around. Also, I'll send out a short survey after the session is done. And so we really do appreciate you answering that, because we do like to get the feedback and how we can improve for next time. And just so you can see who will be the presenters today. So we have Jitendra, who is our soil fertility specialist, Anne, who's our soil management specialist, Hannah Fraser, our entomologist for hort crops, Katie, our plant pathologist for hort crops, Kristen Obeid, our weed management specialist, and myself, Denise, I'm the crop protection specialist, so I deal with invasive species issues and pesticide issues. And then we have a guest speaker, Elizabeth Buck from Cornell. She's a fresh market vegetable specialist. So unless there's any questions so far, I don't think there are. So I'll start with the first presentation. So why is IPM important? And so just, let's make sure okay. So there are various pests that can affect or attack crops. And so we use the umbrella term pests, and that can refer to insects, nematodes, weeds, plant pathogens, and even wildlife. So some people think of insects when we hear the term pests, but there are other types of pests that to keep in mind. In integrated pest management, or we use the acronym IPM quite a bit, So what does that mean? So IPM is a decision-making process that uses all necessary techniques to suppress pests effectively, economically, in an environmentally sound manner. And it's based on research. So there are different ways that you can manage pests. There are biological ways, so you can promote beneficial insects or mites. There are also cultural methods. So for site selection, you may not want to plant a crop if there's poor drainage. Crop rotation you can use, or the grower can use, resistant varieties, certified seed. So you're not bringing in viruses, which tend to be an issue with seed. Planting date, so the grower is planting the crop when maybe certain insect pests aren't present anymore. Sanitation, just making sure you get rid of like infested fruit that could infest the rest of the crop, or if there's a disease issue, you're getting rid of that. Change irrigation practices, so try not to prolong that leaf wetness, because certain plant pathogens thrive under those conditions, such as downy mildew. There's also mechanical methods, so physical removal. So you can rogue out a diseased plant. You can use mulches to help suppress weeds, screens to keep out insects, netting to keep out birds, and there's also steam sterilization of the soil that helps to manage plant pathogens and soil-borne insects that may be in the soil. And another method of control is using chemicals. So pesticides, or another term we use is pest control products. So IPM requires knowledge and understanding of 5 basic components. So the first one is identification. So you really need to be able to properly identify the life stages of a pest and damage caused by pests. And it's also important to understand the biology and its behavior. It's also important to know what natural enemies are present, because natural enemies can help suppress the pest population. So like for an example, like ladybird beetles or ladybugs can help suppress soybean aphid populations. Also, it's important to note beneficial insects and pollinators, because if a method of control is to apply pesticides, they can affect non-target organisms. So you want to keep that in mind too and know what's present in the crop at certain times. Number 2 is monitoring. So using appropriate monitoring techniques for various pests in the crops you scout. Also, it's really important to keep good records. Each time you visit the field, you'll be recording information like the stage of the crop development, disease severity, population levels of the insect. You may be keeping a field map and record the location of the damage. You may be scouting a lot of the field in a day, and you won't be able to remember all this information. So it's really important to record your observations. Number 3 is economic thresholds, and I'll cover that in more detail in the next slide. And number 4, method of control. So the use and timing of appropriate management tools, resistant management strategies, and calibrating your sprayer. So number 4, like the methods of control, that's really up to the grower and not the scout. And number 5 is evaluation, so following up. So that's where the records from your scouting is really important. So for example, if a grower applied a pesticide to control a certain pest, and then it's showing that that pest is still causing a lot of damage, that grower, that's really valuable feedback to that grower that maybe they need to reevaluate the method that they're using. And so just on thresholds, it's a critical part of integrated pest management. When scouting, there will be some level of insects like pests, and their damage will always be observed. However, not all situations result in yield loss. So thresholds are levels of pest injury or insect numbers that will result in yield loss beyond the cost of a remedial action. So usually spraying with a pesticide. So in other words, the density of a pest, it's reached a point at which management intervention, so if we're using the example of, in the case of a pesticide application, it's at which a management intervention is economically justified. So control measures applied at a threshold are meant to prevent the pest from reaching its economic injury level. So the dark blue line. And the economic injury level is the lowest pest population which will cause economic damage. So some crops have very little tolerance for injury, in which case the economic threshold, so the greeny blue line, or also called action threshold, and the economic injury level are quite low. So thresholds may change depending on the crop, variety, and the stage of the crop development. So scouting is key to successful IPM. You need to communicate your monitoring results clearly, timely, and accurately, or else the grower may miss a window of opportunity to use a control method, or may use one when it's not needed. So, and also your scouting, it should be kept confidential. You don't want to be sharing another grower scouting information with anyone else, like another grower. It should be kept confidential. And just why IPM is so important. So the grower is aware of what potential crop management problem they may have in a field or orchard, vineyard, whatever their crop is. And also to control pests only when needed. So this will help reduce the number of pesticide applications and also help with accurate timing of those pest management tactics. It can also help minimize crop losses, reduce risk to the environment and people, reduce risk of disease or resistance development, and it's just good record keeping and like good due diligence. And so you can go to our OMAFRA crop IPM website to get more information on pest management and soil diagnostics for most of the crops that we grow in Ontario, well, hort crops. And it'd probably be a good idea for you to subscribe to our vegetable and fruit blogs to get timely information on pests and pest management. If you're scouting other crops, such as specialty crops, greenhouse crops, tree nuts, we also do have blogs for those, and they were included in the handout that I provided to you. And our vegetable specialists produce a vegetable crop report, which is a weekly update that includes crop updates, weather, growing degree summaries for various vegetable growing regions. And just another example from the On Fruit blog, our OMAFRA specialists produce fire blight prediction maps as well. So they may be useful tools to use. So you'll probably be doing a lot of driving this summer, so you may be interested in listening in on What's Growing On podcast that OMAFRA hosts. And so that's it for me. And I can pass it over to Katie. Is there any questions so far?
Nothing in the chat, but feel free to unmute yourself if you have any questions. Otherwise, I will share my screen. I hope my internet stays stable. Okay. Hopefully that works. Let me know if not. Put it into presentation mode. All right, good morning, everyone, and thanks for being here. So, you know, we did this last year for the first time on Zoom, and I think we were all hoping that this year we could be in person, but, you know, as it goes, we are back with Zoom. And this year, I'm going to go over plant disease causal agents on this webinar. So I think I have about 45 minutes. We'll include some polling questions as we go. So be ready to answer those, and then we'll have some time for questions at the end. So my name is Katie Goldenhar, and I am the pathologist for horticulture crops with OMAFRA. I'm based in Guelph, out of the Bovey building, but of course still working from home now. And so I really hope, no matter what your level of knowledge is here with pathogens, you can find something useful or use this as a refresher, because I think it's really important to go over some of the basics of diseases and the different groups of pathogens so you know what you're looking for in scouting. And also from a grower's perspective, it's really important to know what kind of pathogen is causing their disease, because management strategies are often very different for these groups of pathogens. So let's get started with a question. So I'll get Denise, if you could launch the first poll question. I just want to know a background of how familiar you are with plant diseases. So if you could answer this and put it in, it's anonymous. It doesn't matter if you have none, some, a little. I know some of you have lots of plant pathology knowledge. So you can answer the first one, don't be shy. Just give it a few more seconds. Making sure everyone's paying attention. Looks like we got good response. All right, we can probably end the polling there. Okay, so yeah, most of you have a good amount of knowledge or some basic knowledge, which is great. So hopefully this is a good refresher and some new learnings on potentially some other crops or some other diseases. So I'll just exit out of that. So let's start with the basics of disease. So what do you need to get a disease? It's not just about the causal agent or the pathogen. Now, the definition of disease is the malfunctioning of the growth of a plant caused by repeated irritation by a causal agent that results in symptoms. And so symptoms are typically what you see when you're seeing a disease expression in the field, and it's the plant's actual expression of an infection. And we'll go through some of the common symptoms later on in this webinar. A sign is the actual living organism or byproduct of that organism. And signs are really important when it comes to actually being able to diagnose a disease, because that's a really good way to tell that it's caused by a disease or a specific organism versus some other factor that could be causing it, like an abiotic stress, nutrient deficiencies, or some other pests. Now, as I mentioned, you don't just need the causal agent or the pathogen to get disease. You also need a host. So you need a susceptible host. And so that comes into play, you know, if you are going out and scouting for a specific crop, you know the diseases that affect that crop and that crop is susceptible to. So you already have the susceptible host when you're going into a specific field, but you know, spores are out there everywhere. They're out in the entire environment and they land on a bunch of different crops, but the crop that they land on has to be susceptible in order for it to cause disease. The other one is environment. So you can have both a susceptible host, you can have the pathogen around, but if you don't have favorable environment, you still might not get disease. And so some groups of pathogen really like a lot of moisture, some actually prefer hot or drier conditions. And so that's kind of important to know when you're looking for certain diseases. Some show up earlier in the season because they like cooler weather, some show up when they like warmer weather. So you need all three to get disease, and that's what we call the disease triangle. And so this is just an important thing to remember. You might see reports coming out like in diseases, there's emerging spore trapping. So you might see some results that there's late blight in the area or there's cucurbit downy mildew in the area. But you also have to understand that it also comes into play with the environment. You need those favorable conditions in order to get disease as well. So second poll question, this should be really easy. And I just want to keep making sure you're paying attention. So what do diseases need to occur? So I'll get Denise to launch that second question. So I think that's still number one. Perfect. Okay, so if you can answer question number two, what do you need for diseases to occur? Awesome. I think this is pretty easy, but I just want to make sure that you're paying attention. All right, we can close that one. We have another poll on the next slide. Perfect. Okay, next, a blank is the plant's expression of an infection. So if we could launch poll number 3, is it a sign? Is it a symptom? Is it disease? Or are you not sure? So we'll just give you a minute to fill that in. So the expression of an infection from a causal agent or pathogen. And we do kind of use those terms interchangeably. So sometimes you'll probably hear me refer to it as causal agent or as pathogen, but you know, they are interchangeable when we're talking about plant diseases. Okay, awesome. I think we can end the polling there. So it is symptom. Yeah, so symptom is a plant's expression. A sign is the actual structure of a pathogen or byproduct of that pathogen, and that's a really good way to tell what's causing the disease. Okay, So let's get into plant disease causal agents. So plant diseases can be caused by micro and macroorganisms in all biological kingdoms. So there are lots of different groups of pathogens for plants. And so some of the kingdoms include Monera, which includes bacteria and phytoplasma. So those are pathogens that belong to that kingdom. There's the kingdom Chromista, which has oomycetes, which is another common group of pathogens. The fungi, which I think is the most synonymous with plant disease, includes our mushrooms, molds, mildews, rusts. So that's another biological kingdom. There's the animal kingdom, nematodes. Denise mentioned that quickly before. So nematodes are considered pathogens as well. They cause an irritable stress on the plant, which results in symptoms, and that's the definition of disease. And so nematodes are part of a causal agent or pathogen. The kingdom plants actually also includes plant pathogens, so parasitic plants. Different than weeds, you know, weeds are competing for space, nutrients, and also need to be controlled, but parasitic plants, when they are parasitizing a host to get nutrients for themselves, they are also considered a causal agent or disease, and they cause disease. The other one is protozoa. So there's protozoan plant pathogens, which are slime molds, and you know, clubbered is a really important one, and we'll talk about some of those ones in a second. And then there's the non-living genetic material and proteins, which viruses and viroids fall under. And so those are also considered to be plant causal agents or pathogens as well. So we're going to go through each of these and talk about some of the common diseases that are caused by these different groups here in Ontario, as well as some of the symptoms and signs to look for. So bacteria, like I mentioned, it's in the kingdom Monera. There's over 100 known species pathogenic to plants. See, they're single cell rod, spherical, spiral, filamentous shaped microorganisms. And the thing with bacteria is they reproduce very, very quickly through simple fission. So it's a very simple process, but they reproduce very quickly and can make them really devastating plant pathogens. Now a sign, so the actual bacteria is so small that you can't see it with the naked eye, but they do produce, under ideal conditions, ooze in some cases, and so that's a really good way to tell if you're dealing with a bacterial pathogen, is that ooze, and we'll talk about that in a second. So a really common bacterial disease is fire blight on apples and pears, and so this is caused by the bacteria Erwinia amylivora. And it's ubiquitous across all apple growing regions and in Ontario, and can be quite devastating if not managed properly. It infects the flowers, it infects the vigorously growing shoots, and if not managed, it can lead to branch death, it can go down to the main trunk and actually into the rootstock, and can lead to whole plant death. So this is a really important one if you're going to be scouting in apples and pears to keep in mind. And because it's a bacteria, it's managed differently than you would something like a fungi or an oomycete. But what's interesting here is you can actually see that there's a little bit of ooze production that's coming out, and that's actually how it gets transmitted to other flowers, other shoots. But that's a really good way, if you're scouting and you see that you can confirm that that is caused by a bacteria. So another common bacterial disease is halo blight on snap beans, and so this is caused by a Pseudomonas species. And you can see that its disease name is very characteristic of its symptoms. So this is a symptom here. You have a light green ring around a darker centos, so kind of like a halo. It can infect the leaves as well as the pods, and is quite ubiquitous here in Ontario. Another bacterial disease here, it infects the leaves, it can infect the calyxes, is angular leaf spot on strawberry. And so you can see here on the underside of the leaf, it causes this water soaking, this darkening, which is very characteristic of a lot of bacterial diseases. It causes this darkening of the leaves. And so that's what we refer to as water soaking, is that darkening. And so here with angular leaf spot, like its name, it creates these angular spots and can be quite devastating if not managed properly. So this is a common bacterial disease in strawberries. Same name, caused by a different bacterial pathogen, is angular leaf spot on cucurbits. And so I bring this one up because I think, you know, with cucurbits, It's really important to know the difference between angular leaf spot and downy mildew, which we'll talk about later on, because they belong to different kingdoms. So management strategies are very different. And so when it's this bacteria, you have this water soaking on the underside of the leaves. That water soaking will turn to chlorosis and then necrosis, which is the killing of the actual leaves. And so you can see that here, there's parts of the leaf that have died. But this is angular leaf spot on cucurbits. The last bacterial group that I want to talk about, there's a bunch of different species of bacteria that cause bacterial soft rot, and it can be quite devastating to a lot of the storage crops. It's often referred to as a wet rot when these tubers or these onions or some other storage crops go into storage, if they are wounded or if there's another pest that's attacking them. Often bacteria that's found can get into those wounds and can cause this really wet, stinky rotting. And so that's often a common bacterial disease in some of these storage crops. Okay, we'll have another poll as we end the bacterial section. So what is a sign of a bacterial disease? So we'll get that launched. Is it A, water soaking, B, smelly, soft rotting, C, ooze, or D, what is a sign? We'll just give it a bit to make sure everyone's still paying attention. So we're talking about a sign. So remember from beginning, what's the difference between a symptom and a sign? All right, slowing down. Yeah, so most of you got that correct. It is ooze. So ooze is the actual sign because that's a byproduct of that bacteria. And so that's a really good way to definitively say it's caused by a bacteria and not something else. So just try and exit out of there. All right, now we are moving on to another kingdom. Same kingdom, but a different group of pathogens are the phytoplasmas. So same kingdom as bacteria, and they're bacterial-like. They share a lot of similarities, but they have no cell wall, so they're often referred to as naked bacteria. And so they're only found in the phloem, so where the plant transports sugars in host plants. And it's not known to survive outside of the host or the insect. And so phytoplasmas have to be transmitted by insect vectors. And so they're not known to survive outside of either the plant or the insect host. So typical symptoms, and I'll show you on the next slide, include little leaves, polyploidy, which is actually the abnormal development of floral parts into leafy structures. So where a fruit should be, they actually develop leaves. There's flower virescence, which is the green pigmentation of flowers. Big buds, witch's broom are all symptoms of phytoplasmas. So here we have, you can see here in the strawberry plant, this is caused by a phytoplasma. You have where fruit should be, there's actual leaves that are growing. And aster yellows is probably the most common phytoplasma here in Ontario. It has over 300 hosts. And is vectored by the insect leafhoppers. And so when there's aster yellows shown up in a field, the grower is going to be having to manage that vector as opposed to the actual disease, because that's how it's transmitted and spread. Okay, the next kingdom we're going to talk about is fungi. So again, it's kind of the most synonymous when we talk about diseases in plants. It can produce very visual structures, and so that's a way to diagnose it. But it includes molds, rusts, mildews, mushrooms, toadstools, and there are over 10,000 known species that are pathogenic to plants in 4 phyla. So if you've taken any plant pathology, you know that there's the Ascomycota, Basidiomycota, Chytriomycota, and Zygomycota, with the majority of plant pathogens belonging to the Ascomycota and Basidiomycota. All right, I might have lost you if you haven't taken any basic plant pathology, but I think within fungi, it's important to know that they lack chlorophyll, and that's why they have to infect plants, because they have to get that energy. And with fungi, they all reproduce primarily by sexual spores or asexual conidia. And so we'll talk about the complexities of some of those cycles in different diseases. And with fungi, as I mentioned, they do produce a lot of unique structures, and those are signs that we can use to identify a disease on a plant. So the first one I want to talk about is white mold. So it's caused by the fungi Sclerotinia sclerosum, and it has a very wide host range. So it affects all these hosts you see on the slide as well as others. And what's really interesting about white mold is the mycelium causes that white mold that the disease is named after. But when it gets to the end of its life cycle, it creates these sclerotia, which are those dense black structures you see here. And that's how it overwinters in the soil. And then in the spring, when their conditions are conducive, there's a lot of moisture, it produces ascospores, which can then be released and reinfect the flower tissue for the most part. And then it causes this disease again. And so that's a common one on a lot of different crops. Another common issue caused by a fungal pathogen is apple scab. So it's caused by Venturion equalis, and is really important in our apple production for growers to manage. There's very little tolerance because it produces these scabby-like lesions on the apples, which consumers really don't like. And so it's managed quite heavily here in Ontario. It overwinters on the orchard floor in infected leaves. And in the spring, basically as soon as these apples are coming out of dormancy, that pathogen is ready with its ascospores to reinfect. And so it's managed quite heavily as well in the leaves as well in the fruit. So that's an important fungal pathogen in Ontario. So another group of fungal pathogens is the powdery mildews. Now, powdery mildews are caused by different species depending on what crop, but they're all grouped under the name powdery mildew because they're very characteristic symptoms and signs of this disease. And so you can see here a wide number of crops that are infected with powdery mildew. Again, different species, but all similar symptoms. So they all have this white powdery growth that grows on the foliage and sometimes on the fruit. And so that white powdery growth is actually the pathogen. So if you look really closely under a microscope, you can see the spores, which is a great way to diagnose that it's a fungal pathogen, that this is powdery mildew and not something like potentially residue on the leaves. And then some other symptoms include here, you can see in apples, they have curling of the leaves. And then sometimes there is yellowing of the tissue if that infection is severe enough and causes that plant to start to lose some of its ability to produce chlorophyll. The next group of fungal pathogens is rust. So rust is actually really, it's caused by a bunch of different species, and the species life cycles are actually typically quite complicated. But when you're out scouting in crops, rust really does appear quite similarly. It causes these little red pustules in most crops earlier in the season, so earlier in its infection cycle. But what it actually does is when it gets later into the season and it's getting ready to overwinter, those red spores turn into black spores, and those are the actual teleospores of that pathogen that's getting ready to overwinter. And so most rusts have alternative hosts to complete their life cycle. And so sometimes when we're trying to control a rust in a crop, that results in us trying to control an alternative host. And so that can be quite important in the rust life cycle. The next group I want to talk about is smuts. So smuts, there's different species, again, in different crops, but they're a really interesting group of fungal pathogens. They're typically systemic within that plant, and so they can survive on the seed or in the seed. And then as that plant is growing and it produces its seeds, they're actually producing balls of spores where there should be seeds. And so instead of so that's the way that it travels in those plants. Last fungal disease that I want to talk about is a new fungal disease in Ontario. And so I just want to make everyone aware, if you're taking the strawberry IPM course, there will be lots more on this disease. But I think it's really important just to touch on this one really quickly. Neopistolopchiopsis of strawberries. No one's given it a better name yet. That's the actual species that causes the infection, but it could have the potential to be quite impactful to Ontario strawberry production. And so if you're going to be scouting in strawberries, I really encourage you to get some more information on this disease and look out for it this summer. Okay, wrapping up fungi. Let's launch poll number 5. What is a common symptom of powdery mildew? So is it water soaking on the leaves, black growth on the underside of the leaves, white growth on the top of the leaf, or angular necrotic lesions? Okay. All right, good. Most people are still paying attention. That is excellent. All caffeinated. All right, yeah, so white growth on top of the leaf, perfect. Yeah, that's a good disease name because it's very characteristic of its symptoms. Okay, the next kingdom we're going to talk about is the kingdom Chromista. And the most common group of pathogens within this kingdom is the oomycetes. And so oomycetes produce sexual and asexual spores, just like fungi, and they're often referred to as fungi-like organisms because they were actually thought to be fungi when they first emerged before we had more information. But they're actually more genetically similar to plants due to their cellular makeup. But when it comes to looking at the diseases, they do share a lot of similarities when it comes to fungal infections. But oomycetes produce oospores, that's their sexual resting structure. And their spores we refer to as sporangia. And so sporangia can infect crops directly, but under ideal conditions, they produce the zoospores. The sporangia contains these zoospores, which if you ever have the chance to look at a video of the sporangia releasing zoospores, it's quite cool. They have 2 little tails that allows them to swim in free water. And so that's often why they're also considered water molds, because they really prefer wet conditions in order to infect through these zoospores. So we'll talk about 2 of the more common oomycete diseases that we get here in Ontario. So there are downy mildews that are caused by different species on different crops, but they're all referred to as downy mildews because they're all genetically related. You know, there's downy mildew of onion, of cole crops, of spinach, of cucumbers, and they are all quite impactful to these crops and can be quite devastating if not managed properly. So make sure that you know the symptoms for the crop that you're looking at when it comes to downy mildews. Now, I think it's also important to talk about when we're talking about oomycetes versus fungi. They can both be controlled through fungicides, what we refer to as fungicides, but there are often different fungicides that target oomycetes or that target fungi. So it's really important to know the difference and know what's causing that disease on that crop. The other one that I think, you know, most people are aware of is late blight. It's caused by an oomycete, Phytophthora infestans, and it's really impactful for tomatoes and potato crops, and it's, you know, the one that caused the Irish potato famine a long time ago. And so if not managed properly, it can cause full crop loss, and it cannot overwinter in Ontario, and so it's monitored for heavily when it comes into the province, because once it comes into the province, it needs to start to be managed through specific fungicides. Okay, poll number 6, we have which disease is caused by the water mold group of pathogens? So is it smut, rust, clubroot, downy mildew, or powdery scab? The water mold, oomycetes, often referred to that way because they really need a lot of moisture in order to cause an infection. Yeah, awesome. So it is downy mildew. All right, next we are going to talk about protozoan plant pathogens. So these are single-celled and they lack a cell wall. And so this group of pathogens have resting spores that can disseminate and infect directly, or under wet conditions, they can produce these zoospores. And we'll talk about some pathogens that belong in this group. These ones are often referred to as slime molds. They're biotrophs, which means they're obligate, so they can only infect living host tissue. And they form these plasmodium within the cells of the host plants. And so I think the most common one within this group is clubroot on brassicas. And this is fairly common in Ontario soils. It can be quite devastating to brassica vegetables as well as canola. And its disease name is characteristic of these the swollen roots, which are called clubs. And, you know, it can result in a lack of uptake of water, nutrients, and can ultimately lead to plant death. So this is a really important one if you're going to be scouting in any clubroot and any brassicas this summer, to be aware of this disease. Another one that I think is really interesting is powdery scab. So powdery scab belongs to the slime mold group. It's not a common disease in Ontario, but if you're, if you have any potato seed, it's important to look for these kinds of symptoms because it can survive for long periods once it's in the soil. And another important fact about this disease is it can transmit a virus. So this pathogen can actually transmit a virus, which can be quite impactful. There's another one here, this pathogen here on beets. I'm not going to attempt to say it, but it's another one within the slime mold. And that one itself is not really impactful on the beets, but what it has is it can be a vector of the beet necrotic yellow vein virus, which is really impactful on beets. And so, it's important to know if that slime mold is in the soil because that's what can transmit this virus. Okay. Next group, so this is in the animal kingdom, so we'll quickly talk about some of the nematodes found here in Ontario. And there are hundreds of plant parasitic nematode species. There's lots of beneficial nematodes as well that aren't parasitic to plants, but when we're talking about nematodes as pathogens, we refer to them as plant parasitic nematodes. So they're eel-shaped, transparent worm-like organisms. And sometimes, like in the case of cyst nematodes, those females lose that worm-like appearance at maturity. And one thing that's characteristic of all plant parasitic nematodes is that they have a needle-like mouthpart or stylet, and that's what allows them to actually feed off the plant. So that's characteristic of all plant parasitic nematodes. They have this stylet. And you can see that here in the picture here. If you look closely, there's that little stylet within that nematode. So a common one found in Ontario soils is root lesion nematode, and it can infect a very wide range of host crops. And in some cases, it makes them more susceptible to other diseases. In some cases, it has a direct effect of reducing their ability to uptake any nutrients or water from the soil and can just generally weaken the plant. Another common group of nematodes is the root knot nematodes, and so like the name suggests, it causes this knotting on a lot of the roots and can be really impactful for below ground crops such as carrots when you're actually harvesting the marketable portion of the crop that's below ground. That's where these root knot nematodes can be really impactful, but in high densities they can cause reduced uptake of nutrients or water and reduced overall plant health in any crop. Root cyst nematodes, there are a few in horticulture crops. If you work in field crops as well, soybean cyst nematode is quite impactful, but there are a few in horticulture crops to be aware of if you're going to be scouting something like sugar beets. Sugar beet cyst nematode can be really impactful. We aren't aware of its current presence here in Ontario, but something to definitely be on the lookout because it is quite prevalent in Michigan. And then finally, bulb and stem nematode is another plant parasitic nematode that can be quite impactful on garlic crops or other bulb vegetables. And so it can survive, those nematodes can survive within the cloves, and then when they're planted into the ground, then they can survive in the soil and reinfect any healthy cloves or healthy plants. Okay, poll number 7. What do all plant parasitic nematodes have in common? So A, females lose their eel-like shape of maturity. B, they have a stylet. C, they are all economically damaging to all crops, or not sure. So I'll just give you a couple seconds there. I think most of you got it. Yeah, awesome. So yeah, so they do, so that's the main characteristic that they share, is they all have a stylet, and that's what allows them to be parasitic to plants. Now, you know, some are definitely economically damaging to a lot of crops, but you know, there are different threshold levels. Some crops can tolerate more than others, and so they're not damaging to all crops. And so it's important to know if there's certain nematodes on the crop that you're scouting that can be really impactful. Okay, viruses. So viruses are non-living in a biological sense. They contain only genetic material, so either RNA and DNA. And you know, we all probably know a lot more about viruses now after living through over a year of the pandemic. But within plant pathology, viruses are really can be really important too, and are often considered in emerging diseases. It reproduces and survives within living organisms, and some can survive outside of a living organism, you know, either on equipment, on surfaces, on seeds, in soil. And the only way to really confirm if it's a virus is through a laboratory test. Now you can look for symptoms of a virus, but there's not going to be any visible signs that you can see to confirm if it's a virus. So a really impactful virus that happened in the early 2000s in Ontario was the plum pox virus. So very devastating to growers of stone fruit. And so it caused this ring-like spotting on the leaves as well as on the fruit, which is why it was so important to manage. And so this was managed through a quarantine and disposal of a lot of plants in order to remove this virus from our area. And it was successful, and we haven't had any cases of plum pox virus in a long time. But that was a really drastic measure, but necessary to remove this virus from the production system. Tobacco mosaic virus, I just want to touch on, it was actually the first virus as it was identified. It creates this light green coloring and this mottling in the leaves and this kind of crinkling. And that's a very common symptom of a viral infection, is this mosaic pattern on the leaves as well as this crinkling. But again, this is just a symptom. In order to diagnose it, it would have to be a laboratory test. Another common virus is potato virus Y. Doesn't just infect potatoes, but is really impactful in these production systems. And so just something to look out for, these symptoms of the mottling on the leaves as well as the crinkling, and to get that diagnosed if that's present in the crop. Strawberry viruses, there are lots that infect strawberries as well. In any kind of perennial crop system, there may be viruses present, but they're not expressing themselves. And so it's important when they start to express themselves later in that crop's life, because they can be impactful on the plant health, on yield loss. And again, you kind of see that discoloration of the leaves. And, you know, a lot of virus symptoms can be misdiagnosed. They look sometimes a lot like nutrient deficiencies, and you'll get some information on nutrient deficiencies later on in this presentation. Last one I want to talk about is a really impactful disease for the greenhouse sector, and you've probably heard of it if you work with any of those crops, is the tomato brown rugose fruit virus. And you know, this one really persists for long periods of time and is quite easily transmitted by humans and by equipment. And so with all the different touch points when you're working with these crops, it's really important for disinfection when it comes to this viral infection. Last poll question, I promise. Can you answer this one? Viruses cannot survive outside of a host. Is this true or false? A little bit of confusion here. All right. Yeah, so the answer is false. So viruses can survive, they cannot reproduce outside of the living host, but they are able to survive on equipment, on seed, and so that's really important when it comes to transmission of viruses, to know that they can survive outside of a host. So last group of pathogens that I quickly want to touch on is parasitic plants. And so there are a number of parasitic plants that can cause disease, and why they cause symptoms because they're actually parasitizing the plants to obtain nutrients. Some need all their nutrients from other plants and some just need some. So again, some produce chlorophyll and only depend on other nutrients, and some really don't produce chlorophyll, and so they need that plant to live off of. And so here are some examples of parasitic plants. You have dodder, dwarf mistletoe, witchweed, all which are parasitic plants to some crops. Okay, finally, just ending with some resources. So when it comes to diseases, and now they've actually started to include some insects as well. The APS compendium series, which is linked here, and you should have that in your notes. If you're working with a specific crop, these are really helpful. It lists all the different diseases that have been known to that crop in North America. So some, are specific to the States. But I think when it comes to horticulture crops, these can be really useful for a resource. Other resources, these are more for management of diseases, but these can be useful if you're in a specific crop and you want to know the different diseases that are present in that crop. You can use these OMAFRA publications, these crop protection publications, to kind of as a reference for some of the common diseases. And then there's a list of other ones here that I found particularly useful for horticulture crops and for some basic plant pathology as well. And then just some online. Denise mentioned the Ontario Crop IPM. There's other fact sheets when it comes to some of the other diseases that might not always be captured. There's the OMAFRA specialist contact information. You're always welcome to reach out to us if you have any questions. And then there's the horticulture blog. So Denise mentioned the fruit one, the vegetables one, but if you're working in specialty crops, floriculture, greenhouse, or nursery crops, those are all also available as blogs as well, and so you can subscribe to those as well. With that, there's my contact information. I think we have some time for questions, and I'm always happy to chat about diseases in horticulture crops. So with that, I think I will stop sharing and open it up to any questions. Everyone's ready for more coffee. I'll be here the whole time too, and we'll chat a bit more later. So if you have any questions as we go, feel free to send me a message and I'll answer that in the chat. And this picture behind me, I should say, is from phytographics.com. It's a very cool website that's been put together with a bunch of different diseases and their pictures, and they're quite beautiful. So maybe I'll put that in the chat if you want to look at some beautiful disease pictures if you're a pathologist nerd like me. All right.
Okay. Well, thanks, Katie. If there's no questions, I guess we can break, and gives you a little extra time. So try and be back here for 11:10, and then we'll have Elizabeth Buck speaking on taking good diagnostic pictures. Okay. Okay. So welcome back, everyone. Everyone should be coming back for 11:10. And our first speaker is a guest speaker, Elizabeth Buck. Elizabeth is a fresh market vegetable specialist with Cornell Cooperative Extension's Regional Cornell Vegetable Program. She assists growers of all sizes, experience levels, and production style in 6 county regions in Western New York to solve their production challenges in crops ranging from asparagus to zucchini. Elizabeth's special programmatic focus areas include weed management, soil health, and climate resilience, and vine crops production. So I'll hand it over to you, Elizabeth, and thanks for being here.
Yeah, thanks for the opportunity to come and speak to you guys. I am, like Denise was saying, I'm a fresh market vegetable specialist. My job is really wide and varied, and in my work, I'm constantly taking photos. They're photos for me, for my records, to use in future presentations for teaching, They're photos that I'm sending off to our pathologists and other research personnel at our Cornell University for consultation. I'm taking a lot of photos that I'm sending to the growers to let them know what's going on in the field, or receiving photos from the growers. So everything I'm going to talk to you today is based off of that experience. I am going to drop into the chat, if I am good enough here. I'm going to drop into a chat a link, and that is where you can find a PDF that will summarize everything I go through. So if you miss something, just go and check out that PDF. So let's get started. Diagnostic photos, I understand that today this is sort of a whole diagnostic training day. They're really useful in communicating what is this to the grower, communicating it to whomever you need to. And it's all about capturing the right image. And this first image I have here, I enjoy it because you see the corn's not doing so well. This is a field of sweet corn. And the soil is a really ridiculously light color. This should be a dark silt loam soil. And in this whole area where the crop is doing poorly, turns out they had dumped a load of lime there about 5 years ago, and it's still carrying for infecting the crop. So image is worth 1,000 words, right? So let's go through this. I cover a huge territory, lots of different crops. Photos are just key for communication. That's just the point I have to put this slide in. My Dama doing a presentation. So we got that box checked off, right? When we're talking about photo taking, there's a few things that you really are going to be helpful to know. One is that cameras sense far more light than our eyes can. Our eyes saturate at a much lower photon flux density than what plants feel and sense, and definitely lower than what a camera senses. So that's why when you're taking photos in the summer, so very often the image appears washed out, appears like that image on the left, okay? That's because the camera is sensing the true amount of light. We are incapable of doing that. Second thing, in diagnostics, color is key. I had a conversation, long conversation with my tech just the other day about, you know, we were talking about how it affects our jobs and how subtle differences in color, developing the skill to see that is really underlies so much of the disease and nutritional and viral diagnostics that we do. And so if the photos you're sending in are not true to the color that's presenting in the field, that's going to influence someone's interpretation of the image and what the problem is. So in these three panels, we see the one all the way to the left with that is the true color. We know it's the true color because the dollar bill is the correct color. Maybe not as useful for you guys, but that's the right color. The one in the center is too blue. That's what it's going to look like if you're trying to use filters on your camera. You're going to change the color. You can make it too blue or too yellow. The other blue and yellow, most shades, most sunglasses are going to have either a yellow filtering or blue filtering, and you get that sort of color distortion when you're wearing them. If you have shades on and you are seeing crops that look more blue or more yellow, you're not going to get the same level of information as if you have the true color on it, and you need to make sure that carries through into the photos. The third thing is, I don't care how good you are at taking photos. You can take beautiful, wonderful images, win prizes at, you know, conferences, get 100 likes on Insta. If you aren't capturing the right part of the field or the plant, it's worthless for diagnostics. So looking at these panels here, this is from a potato call I took last year, and you can see a large chunk of the field was just smoked, and it switched right dead on with the variety change. And so we were thinking, oh, like variety change, bad seed, something like that. And as we come over, we're seeing a section of that same variety that didn't get smoked, and it's in the shade. So we were really trying to sort out what happened here. But we can't do it from these photos. You know, if I had just received these photos from a grower, I would say, oh, you had bad seed, maybe something's going on. I wouldn't have picked up what actually was happening. I had to dig for that, right? So you have to know what you're looking for. You have to have some sense of what the problem may be in order to get a diagnostic photo. It's not a photo that's going to solve everything, give you diagnostics. You have to have that skill and that, go through your checklist, go through your, is it abiotic? Is it biotic? Is it likely to be soil caused or aerial caused or man-made before you get to the point of taking photos? Because really, I need to get down to the fact and see that the roots on the healthy variety were just fine, And the sad variety, they were not even starting to set stolons. And that is what the appropriate diagnostic photo was. Make sense? Used to teaching live, but please throw things in the chat. I appreciate interruptions. I want to make sure things are clear. So I'll pause for a second, give you a chance to type anything into the chat. Okay, not seeing anything come in. Let's go into some tips for actually getting good diagnostic images. So the first one is that you want high quality images. What constitutes a high quality image? That's going to be, one, the image matches the eye's perception. Two, the image is sharp. And by sharp, I mean it's crisp, it's clear, it's focused. And three, it captures the correct subject. So if we look at this image here, anyone know what this is? You shout it out, pop it in the chat. Any idea what this is? This is how I make sure you're there, you're out there and you're awake. Anyone scouting vine crops this summer?
A quiet group today.
I know, come on guys, I'm not moving on until someone answers. Cornell, we do participatory learning. Aphids with eggs. That is often what people will think this is. Eggs, yes. Aphids, no. Anyone doing vine crops? Squash bug. Yes, these are squash bugs. The correct subject here is not the leaf. The correct subject are the little bugs. And if you look close, they have the big long legs, but they also have these little green butts on them at this stage. Okay, and these coppery eggs, you can see that the color is darker in the back and lighter in the front. That's the difference between ones that are hatching and not hatched. Well, obviously the hatched ones are crawling around out there, but you can tell how close they are to hatching based on the color. And if you really got a sharp eye, there is an aphid in the image. You gotta make sure you capture the correct subject. And knowing in this case that it was the insect that I needed, not the eggs. I needed to really see that body on it to make sure I don't have aphids and send that off and be able to send it to someone like the OMAFR specialist or your boss who could help you out with that. So quality tips, very basic. The first one, clean off the lens. I don't know if you're like me, my phone is constantly getting dusty. I've got a case on it, but meh, it only helps so much. I'm clumsy. I stick my finger over the lens. It gets all smudged. It gets mud on it. Just stop. If you need to send off photos because you're not sure what's going on, just take a moment and clean off the lens. I mentioned it earlier, filters, they mess with the color scheme. They mess with the sharpness. They're more trouble than they're worth. It may seem like it helps, but if you flip your phone into manual mode, your camera into manual mode, which I will take you through the settings, you can do a much, much better job than filters, and it's faster. All right. So other tips, you want to clear out distractions, anything from the background that doesn't need to be there. In this case, this is a very sad little kale plant. It has downy mildew. There were a bunch of weeds in the background, so I just took a moment and yanked them out of the way so I could isolate the subject. And then the second thing is, the camera's having a really hard time focusing. It's small, it's close to the ground. I don't have a lot of other things to help it figure out what it wants to focus on. And that's where you can flip your settings into portrait mode. A lot of phones have portrait mode, and it just gives you such a better crisp image. And this is literally the difference between not portrait mode and portrait mode. And you see the veins pop out, you see the dark blackening underneath pop out, you see the color patterns come. It's the best trick I know for getting something to focus where you want it to. Getting back to the color thing, this is that same squash bug image that I showed you before, and now it's all washed out. This is when I wear my shades, I tend to, it filters out more of the blue, so I see things in a more yellow mode with my sunglasses on. This is what I would see in the field if I am trying to take a picture to match what I'm seeing. That is always your goal. Get the image you capture to match what you're seeing, especially for color, but also for crispness and other details. I would have taken an image that looked like this, And that's not very useful because this whole plant looks chlorotic and just, this is not the color of happy squash. You want to take the shades off your face, then you want to put shade over the subject. If it's really bright and sunny, you can cut out a lot of that washing effect by shading the what you're taking a photo of. One of the hardest things is when I get a diagnostic image in from a grower and they're standing in the field and they're standing there and they just look down and they point and shoot, and half of the image is in shadow and half of it's in the sun. I can't see any details in the shadow part, so it's very hard for me to go and say like, hey, it looks like you might have some verticillium wilt going on there, or hey, yeah, that looks like you have strip scarring on your peppers. That's difficult to do. So by putting yourself in a position where you're shading, you're casting a full shadow on what you're trying to take a photo of, is going to make your job a lot easier because you won't have to mess with adjusting the white balance. You're taking out a lot of that overexposure, and it also gets you to stop and think about, how am I going to shade this? How am I going to stabilize this image? Okay. Stability of the camera. I'll show you an example earlier when I just anchored it or not. Stabilizing the camera isn't hard. You know, prop your elbow on a tailgate, on your knee, on a post, the ground, your boot, whatever it is, just help get things to stop shaking. The smaller something is that you want to take a photo of, or the more the object is moving, the more important it is for you to be stable. You know, if you have 1 point of stability, you're gonna get a decent image. But if you have, say, 3 things moving around, there's no chance. You have no chance of getting something that's crisp. Because of that, you want to remove wind as a factor. Lots of times when I'm in the field and I'm like, you know, putting shade on it, you know, kneeling down and putting the subject in the shade, oftentimes the sun is from the south or the west in the middle of the summer. That's also the direction our wind comes from. So it's a twofer. I'm blocking the wind, removing that as a factor, which increases the stability, and I'm correcting for the overexposure just simply by where I'm placing myself. And then bugs, notoriously difficult to take an image of. This is a western bean cutworm that's going to go after your dry bean industry. That's going to go after dry bean, snap bean, corn, maybe a couple other things. These guys are very hard to take images of because they fly around. Pop them in the freezer, pop them in a fridge, keep cooler in your car, throw them in there for a half hour, 45 minutes. They're gonna they're gonna cool down, slow down, then you can get a nice picture of them. So any bugs that are just a pain in the butt, grab the leaf that they're on, stuff them in a bag, take them home, toss them in the fridge, toss them in the freezer for a half hour, then you can photograph them and they won't be running around so bad. It's really tempting and easy to zoom in on an image or on a subject, to zoom in on it. What happens when you zoom in is that you are essentially changing the pixelation, right? And as you, just like if you take something on the computer screen and you expand it out and blow it up, it gets fuzzy. The pixelation doesn't work right. When you are working at getting something small or adjusting something, I've found that I get better quality images if I move the camera physically closer or further away from the tiny thing I want to take a picture of, and then zoom second. A couple reasons for this. One, anytime you move the camera around, you're going to change where it's trying to focus. Every time it changes where it tries to focus, it also resets the white balance, and you have to go through and manually slide that little light bulb or that little sun slide bar up and down and adjust that again. So first, set your distance, then you can adjust the white balance, then you can zoom from there. And that is A, more efficient, B, gives you better quality image in the top. This was me just trying to zoom in and get a picture of some fungus. If you have something really small you need to ID, I really like using a hand lens. They sell little attachments that you can put on smartphones that are like little microscope or macro shot attachments you clip over the camera. Again, I'm in the field, the phone's in the case, and it doesn't work so well. I always have my hand lens with me, and you guys will too. So pull out your hand lens, clean that off, get the fingerprints off of it, and put your hand lens over the camera, and then use those two things together to get the shot you want, or hold the hand lens over whatever you're taking a picture of. And you can get much, much better detail on fungus. So if you're trying to do a field ID of, say, downy mildew or late blight, or I don't know what attacks corn, you want to tell the difference between like northern corn leaf blight and some other gray mold of corn, or clearly on your vegetables, use your hand lens. It helps tremendously. I mentioned adjusting the white balance. That's just how much lighting exposure is. Talk about using shade. The other thing is to take high resolution shots when you can. These are two caterpillars, or this is the same caterpillar. I took the first series of shots just regular. The second series of shots, all I did was stabilize the camera and change the resolution to high resolution. So I was just taking them real quick right after the other, and you can see the difference. It's more important to use the high resolution if you know these photos will go into any sort of publication or internet, or they're going to be emailed, because when you compress images down, you lose data. So start off with the most data that you can, and that will maintain your image quality as it goes through those various compressions. And that's it in terms of the actual tips and tricks. I know I'm moving fast. Check out the PDF I posted. I do want to real quick just flip over to a case study I had this week, if I have sufficient time. Is it okay to do that, Denise?
Yep, you have time.
Okay. Let's see, you guys seeing a geranium pot?
Yep.
Cool. Got a call. Actually started off with a photo. Grower sent me a photo, and there were some sad looking transplants, asked me to come out. This was Friday, and didn't know what was going on. Thought maybe there was some sort of nutritional problem. Walked into the greenhouse, and it's primarily in eggplants. It is distributed. He's got transplants for about 4 or 5 acres of eggplant. The symptoms are distributed randomly and evenly throughout the transplant flats. They are not grouped up into any one corner. There are a couple tomatoes showing it, and in another greenhouse, there are some geraniums showing the issue. And the question was, what do you think is going on here? How bad is it? Is it viral? Is it environmental? Is it nutritional? And start off with the eggplants, because that's the most clear, and seeing the distribution of it being random. And I had to ask the question of, okay, if I'm seeing this in greenhouse 1 with your eggplants and tomatoes, how am I also seeing this in greenhouse 3 with your geraniums? And the answer was, well, they used to live right next to each other, all in greenhouse 3. They had been moved. And this goes to getting the right diagnostic imagery. If I had just stuck with the eggplants and the random distribution, it could look like something that was seed-borne. By asking the question of, have these always lived here? How come I'm seeing it in 3 different crops in 2 different greenhouses? It's important to know that they were all right next to each other in proximity, that the likelihood of having insect vectors means I need to check 2 houses for insect vectors instead of 1 house for insect vectors. And that the random distribution I thought I was seeing may not actually be random. It could have actually been a pattern because the trays all got rearranged. So knowing that, I went through and got the photo. So there's your first thing, like always ask the question so you can try and get the right image. Talking about quality of images, yeah, this is a fine image, but I could look at this first geranium on a glance and say, okay, I got red along the outside edge, I have discoloration, interveinal chlorosis. This is gonna be something nutritional. Because the images in the back is clear, it's just kind of like a quick snap, right? If I actually stop and take a better quality photo, and this is not the best quality, but if I focus in on that young growth for virus, we're looking at newer tissue having most of your symptoms. I'm saying it's not interveinal chlorosis. It's that the leaves are disfigured. They're not expanding out correctly. They're blistering. We have odd modeling. And it gets worse as the plant gets younger. So the difference between these 2 images is, one, I could send it to someone and get off on the wrong track of chasing a nutritional issue and waste time, when we need to be chasing a viral issue and eliminating these plants from the greenhouse and following up with dealing with the vectors. Talked about using portrait mode. I was trying to get a shot of this eggplant seedling. Regular snap and shoot wasn't working. There was too much depth behind the subject. So I just flip it into portrait mode. Tried to lay it down first, still not really working because the plant is 3D, right? laying it down on a flat surface doesn't always work. You can't capture 3D. Instead, put it in portrait mode, you keep the 3D definition, and you get a good example of the viral issues. Okay, so we got strapping on the leaves, deformation, curling, crinkling and blistering, and then trying here to get this, which is, see how that vein is all twisted and drawstringed in? Point being, it takes several images to get one or two that you want, and that's okay. If you're taking diagnostic images, take your time. I usually spend 5-10 minutes. If it's a problem I know I'm going to send off to someone, I spend 5 or 10 minutes just taking photos to get 2 or 3 photos, but it's okay. It's worth it, because what happens is I send the photos in advance with an email or a text saying, hey, I'm sending something to the lab. Here's the images. When it gets to the lab, they're going to check it out and double check, but most of the time off the photos, they can give me something they're 85-90% sure on, and we can start working on a solution for the grower. We don't have to wait the week for it to get to the lab and run through the full diagnostic test, because if I can send a good photo, I can start someone off on a better path, or I can at the very least tell them what not to do. And that's all I got. Just be patient, take several images, play around with it, throw your phone in manual mode. Thanks. I think I'm out of time. Please send me your questions. I'll answer them in the chat.
Yeah, there was one question.
Okay, is it better to change the camera settings to get an accurate picture, or change the camera settings? It's always better the better quality you start with, the less time you have later. Yeah, you can fuss around with that on the computer, but A, you have to remember to do that, and you have to remember all the things in the field, and B, that's extra work for yourself when you get home.
Hi everyone, I'm Hannah Fraser. I'm the entomologist for horticulture crops with MAFRA. I normally work out of the Guelph area. I'm housed at the University of Guelph in the Bovey building. And I'm coming to you virtually again this year. So this presentation is about half an hour long. It's an intro to entomology for IPM scout training. I'm going to turn off my camera just so that it's not a distraction while you're listening to the presentation. And it won't, I don't want to cover up any of the content. So I'll turn it off now. Thanks, great. Okay, so again, this session is really not meant to replace sort of comprehensive entomology. I'm really just touching on some basic concepts. My thought is that I want to give you some information that you need for the upcoming crop specific sessions that you'll be attending. And I know some of you are attending a lot of them and some of you are attending just a few of them. And I do recognize that there's a real range of experience in terms of working with insects. So this is not meant to, again, not meant to replace anything like that, but I hope that this will pique your interest and help prepare you for the upcoming sessions. So what I'm going to do is I'll talk a little bit about basic features of insects. I'm going to talk about life stages and the whole concept of metamorphosis. There is a handout that was provided as part of the intro to IPM on temperature and development. I'm not covering that here, but I do have a recorded presentation on that as well. I will be covering the orders of economic importance, and then I'm going to take you through a gallery of symptoms and signs of arthropod injury, just to give you an idea of some of the spectrum of things that you'll find out there. Okay, so the first question I always ask is, why am I monitoring for insects? Well, of course, you want to know who is out there, whether it is a pest species or whether it's a beneficial species. So if you're not out there monitoring, you're not going to know that. One of the great things about monitoring is it can give you some excellent information on what's happening within a population. So is the trend that the numbers are on the increase? Are they on the decrease? And this is really important from the standpoint of the grower, because they need to know whether or not a threshold has been reached and it's time to treat. One of the other things that you'll find by actually going out in the field and looking is to find out what life stages are present. So there's different life stages of insects, some of which may be exposed and vulnerable to pest control measures like an insecticide spray. It's also important too, because with the new insecticides and miticides that are out there, some of them are specific or work better on certain life stages versus others. So that's really important information. In addition, there may be some hot spots in the field where the grower may just be able to do a treatment of one area and not the whole field. Maybe it's just a border or one side of the field or somewhere near a dusty road where there's lots of mites. So that's important information. It'll also tell you whether there's anything that's kind of unusual. And this I find is a key thing that we really need to stress more and more, because there's so many different new pests that are arriving into Ontario, and the earlier that we find them, the better. So you're another set of eyes, and you can help us out in that regard and help the grower out as well. And one of the other things too is if the grower has gone in and made a spray, you are going to be going back maybe the next week when you can get back in the field to follow up and to see what kind of an impact that spray actually had. So was it effective or not? Is the pest still present, and are the numbers still alarming? And apart from that, I think insects are just kind of super cool, and hope you take an interest in them over the course of the summer. So in terms of looking at insects, insects and mites are arthropods. These animals have jointed limbs and mouth parts, segmented paired appendages, and an external skeleton that's made of chitin, and this protects them and it gives them structure. Insects have 3 body regions: the head, the thorax, and the abdomen. The head has a pair of antennae, which are highly varied in form, and these give them all sorts of sensory information. It also has a pair of compound and or simple eyes. Legs and wings are also present on the thorax, which is actually divided into 3 subsections, and each of those subsections has a pair of true legs, with some exceptions in immature stages. Adult insects have 1, 2, or no pairs of wings, and actually the ability to fly makes insect unique among arthropods. Mites themselves are also really an important part of the conversation when it comes to pest management. Pests like the 2-spotted spider mite, which is shown here, are important globally because they really rob crops of yield, and they're prone to the development of pesticide resistance. But as with insects, not all mites are pests. Many are important predators, some of which are critical biocontrol pests. Biocontrol of pests in greenhouse production systems, as well as in the field as well. Mites are also arthropods. They are more closely related to spiders than to insects. They have piercing, sucking mouth parts, basically like a straw to suck out the sap and contents of whatever it is they're feeding on. They have simple eyes, they lack antennae, they lack wings, and the adults can have either 2 or 4 pairs of wings. Metamorphosis is a term that many of you are probably familiar with. Think of the classic monarch butterfly that transforms itself from a caterpillar into a beautiful butterfly, which looks like a completely different creature. Winged insects go through distinct stages of development before they become an adult, and this is referred to as metamorphosis. They grow through a number of successive instars, each of which is terminated by a molt and ecdysis that replaces the old larval cuticle with a new one, and all of this is controlled by varying levels of hormone. Shedding the old cuticle allows the insect to grow. Think about your shoes when you were growing up. Eventually your toes hit the tips and you needed a new pair to accommodate your growing foot. Well, insects go through these successive periods of molting and growth. There are often new features that show up with each new immature stage, such as colors or patterns or external features like developing wing buds. There are also internal changes that you can't see. The changes that occur and the molts are the result of hormones. Changing the levels of these hormones eventually triggers the insect to molt to the pupal or the adult stage. I really love these 2 pictures. They're examples of insects that are just molting or have just molted. So the softfly larva on the bottom is still actually growing. So what you're seeing at the top right corner is the cast skin. It looks like a dead, dried up insect, but that's actually the cast skin. And then the cicada on the top left is just coming out of its it's emerging from its nymphal stage, and its wings haven't expanded yet, but eventually they will, and it'll darken up in color and it'll fly away. There's several types of metamorphosis that occur in insects and their allies, their relatives. The first is incomplete metamorphosis. It does not apply to sort of more advanced insects, but it does apply to mites and to what we call primitive insects. And basically what happens in incomplete metamorphosis is that the immature stages look very, very much like the adult. They just look a little bit smaller. So there's no wings that have to develop here. It's just that the insect gets a little bit larger, or the animal gets a little bit larger each time it sheds to the new stage. Mites are a really great example here. Mites go through the egg, larval, stage, as well as nymphs, and there's multiple nymphs, and then adults. The larval stage has only 3 pairs of legs, whereas the later instars of nymphs and the adults have 4 pairs of legs. And there are some species that only have 2 pairs of legs as adults. But that is, again, what's important here is that there's no wings. There's no kind of major transformational change that actually occurs. Winged insects all undergo either gradual or complete metamorphosis. In both of these cases, there's at least one major transformational change as the insect grows to adulthood. With gradual metamorphosis, each successive molt brings small changes. Many start as an egg, although many aphids can actually reproduce asexually, and they give birth to live young. Nymphs and adults look similar to one another, except the adults may have wings. Older nymphs have external features like wing buds that you can often see. The major transformational change occurs when the last nymphal instar molts to an adult with wings. Most nymphs and adults live in the same environment and feed on similar things. Here are the different life stages of a stink bug, including eggs, the 5 nymphal instars, and the adults. Insects that undergo complete metamorphosis undergo 2 transformational molts. The first is when the larva becomes the pupa, and the second is when the pupa becomes the adult. Larval stages are referred to as larva, caterpillar, maggot, or grub, and their forms are really variable. Some have well-developed heads, thoracic legs, and prolegs on the abdomen. Others lack prolegs. Some lack legs on the thorax. Some don't even have obvious heads. The feeding habits of larvae and adults may be the same, or they may be completely different. Some adults have vestigial mouth parts and they don't feed at all. Common orders you may be familiar with would include beetles, the moths and butterflies, and the flies. I just wanted to mention diets briefly. Insects can be both specialists or generalists, and this applies both to the herbivores or the predators and the parasitoids. Some of them have very, very specific diets, these monophagous insects like the Parassilla that feeds exclusively on pear. And then you have some that feed more broadly on related genera like the Colorado potato beetle that will feed on plants in the solanaceous family, so potatoes and eggplant, as well as solanaceous weeds. And then there are the true generalists that feed basically on everything. And I always like to think about brown marmorated stink bug, which is an important new invasive species that we have here in Ontario, as well as many other parts of North America, and unfortunately Europe. It's really spread. But this insect will feed on many, many different unrelated hosts. So we kind of refer to it as a cross commodity or a landscape level pest. So the different orders of insects, I mean, certainly there's more orders of insects that are indicated here on this list here. The classification is based on characteristic body forms. There's several orders of importance to agriculture that I'm going to go over today. Remember how I said the ability to fly is unique to insects among arthropods? Well, insect orders get their names from a crude description of their wings. Some of the other features you're going to be looking at might be the shapes of the legs or the antenna, but all of this is really beyond the scope of the intro session. I'm going to cover these orders with the exception of Orthoptera, the grasshoppers and crickets, as I suspect most of you know what these look like. Some of the most serious pests globally include members of the Homopteran suborder. For example, virtually all of the crops you will learn about have at least one species of aphid as part of their pest complex. Homopterans are usually small insects, but they are highly varied in form. Sometimes it's even difficult to realize you're looking at an insect. You can see the scales in the picture on the top middle. They look like hard bumps on the plant, but if you flip them over, you'll see the dead female or maybe even a live female scale underneath and all of her babies. Called crawlers underneath. Homopterans have piercing sucking mouth parts, which we refer to as stylets, like a straw. Not all of the adults have wings. Some, like aphids, have winged and wingless morphs. Many are important vectors of plant diseases. These insects have a gradual type of metamorphosis that includes eggs, nymphs, and adults. In some cases, the females give birth to live young, as I mentioned with aphids. The Heteroptera are often called the true bugs, and they all have the bug in their name. If you look at that, like stink bugs and plant bugs or minute pirate bugs or mullein bugs, word bugs, so true bugs. If you look at the forewings of these insects, you're going to see that the top part of the wings are leathery and the bottom part of the wing is membranous. And then the forewings are also membranous. This group of insects can be plant feeders or predators or both. Some will also attack mammals, birds, and other animals. They have piercing, sucking mouth parts, a prominent beak in the front part of their head that kind of hangs down like a sword that's slung back under the head and body. These insects also undergo gradual metamorphosis with egg, nymphs, and adult stages. And I just have a picture here with two stink bugs. One of them is predatory and one of them is plant feeder. If they have a kind of long, narrow, spindly looking mouth part or beak, proboscis, they are likely plant feeders. And if it is robust, they are likely predators. Most Lepidoptera are plant feeders. They include the moths, the butterflies, and the skippers. One of the important features to help tell whether you're looking at a moth or a butterfly or a skipper is the shape of the antenna, whether they're knobbed or thread-like or leathery. The adults have a coiled proboscis, which is used to siphon nectar or other liquid-based foods. Although some don't feed at all, some may just have this vestigial mouth parts. The larvae, though, they all have the chewing mouth parts, and they're really the damaging stage that we find in agricultural crops. I love this picture of this caterpillar here. I think it shows the three different body regions really, really well for this larval stage. So you can see the head, you can see the thorax with those true legs on it, you can see the abdomen, and then there's what these call these little fleshy abdominal prolegs on the abdomen. If you're looking at a Lepidoptera, they will have between 2 and 5 prolegs on the abdomen, and all of those prolegs have these little hooks on them that we call crochets. And these are important features because they can be confused with sawfly larvae, which we'll get to in a few moments. Beetles are a really important group of insects, and many of them have been described. Both adults and immatures have chewing mouth parts. This group contains many pests, but there are also many beneficial insects in there, like lady beetles. And many of them are also decomposers and scavengers. The adults have 2 pairs of wings. The forewings are hard elytra that protect the membranous hind wings. So I've got the little arrows showing those little protective elytra that are there. And if the elytra were closed, they would meet in a straight line down the back of the insect. And then the hind wings are membranous. Again, these are really varied group. Some incredibly beautiful insects belong to the beetle order. And these undergo this complete development of egg, larvae, pupae, and adults. These are some of the larvae of the beetles that you'll find out there. And you will find the immatures in crops as well as the adults. Their forms, again, they're highly varied, but they all have well-developed head capsules. Some of them have well-developed true legs, so those legs on the thorax. Others don't have them. None of them have legs or prolegs on their abdomen. So that's an important distinction for them. So Hymenoptera are another important group of insects that play different roles in agricultural crops. So when you think about bees, they're beneficial. They're incredibly important in terms of pollination services. Ants are real ecosystem engineers. And wasps are actually important predators and parasitoids. The sawflies, however, are generally pests. Hymenoptera also contains solitary as well as social species. The adults have 2 pairs of membranous wings and large compound eyes. Females have an ovipositor or a stinger. Ants, bees, and wasps have a narrow waist, whereas sawflies are thick-waisted as adults. Most of the time, what you're going to be seeing when you're scouting are the adult forms, with some exceptions, like with the sawflies. Sawfly larvae can look a lot like caterpillars with a well-defined head capsule and true legs. They also have those prolegs. So caterpillars, remember, they can have up to 5 pairs of prolegs, where sawflies have 6 to 9. They have 6 to 9, so considerably more. So count those prolegs, know what it is you're looking for. And one of the other things too is their prolegs do not have those little crochets, those little hooks. And sawfly larvae, they are often exposed feeders, but sometimes they feed in sort of these groups of them, or they may be solitary feeders. Sometimes the eggs are laid within the plant material as well. So this female here is laying her eggs inside the needle, and you can see all the little pale dots where the eggs have been laid here. So Diptera is another incredibly important group. They are diverse and they contain both beneficials and pests. So this is a common theme that you're going to hear through this, is that yes, there's a lot of pests in these groups, but there's an awful lot of beneficials as well. The adults can have variable different mouth parts. They can be sponging or sucking or piercing mouth parts, really vary depending on what it is that they're actually feeding on. The larvae technically they have chewing mouth parts. They do have mandibles, although they're often modified, or they may have mouth hooks. So typically when we talk about their feeding, we talk about rasping or maceration. So one of the things with Diptera is that they have one pair of wings, plus they have these little halteres, little tiny, they look like little tiny wings, and they're important for stability and maneuverability. So if you watch flies moving through a system, like the adults flying, you'll see that they're real acrobats. They can change direction very, very quickly, and that is because of these incredibly maneuverable wing system that they have. And so these insects too also have that complete development going through egg, larvae, pupae, and adults. So if you look at the larvae, they're often referred to as maggots. They lack true legs. Some have well-developed, so no legs on their thorax. Some have well-developed heads with chewing mouth parts. Others have, again, these reduced mouth parts, including those hooks. Most of what you're going to see in the crops that you're working with are kind of the classic maggot, which is like no distinct head and they have mouth hooks. Or if you work in the greenhouse, you might find some that are more gnat-like, that have a head capsule that you can actually see. So the last order that I wanted to cover are the thrips. Adult thrips are really delicate looking insects with these fringy wings. Most of them are quite small. These insects have rasping style mouth parts, and when they feed, the injury looks a bit like silvery streaks on foliage or discolored fruit surfaces. About half of them feed on fungi. The rest of them are plant feeders, but there are some predators in the mix as well. So the injury can be the result of the feeding by the adults and the immatures, but it also can occur when the female lays her eggs in the plant tissues, and when those eggs hatch and they come out, it actually injures the plant material. So these guys have kind of an interesting form of metamorphosis that includes eggs, 2 larvae, 2 pupae, like a prepupa and a pupa, as well as adults. And this is just a picture to give you some perspective on size, because these thrips are within the flower head of a strawberry. So you can actually see how small they are. And they are flower thrips. They love flowers. Makes sense, right? So we have talked about symptoms and signs of different pests that are out there, whether you're referring to diseases, and this also can apply to insects as well. So symptoms are changes in the plant growth or appearance. So missing plant parts, clipped shoots, dieback, distortions in tissues, spots on leaves, holes and tears in leaves, or chewed leaves, or discolored leaves. And some of the signs, these are the evidence of the actual damaging factor. So that can be the insect or the mite itself, the cast skins, the frass, which is insect excrement, the honeydew. A lot of those homopterans produce a lot of honeydew, which is sticky when they feed. It kind of comes out the other end. And a lot of other insects like ants and wasps are attracted to that. It also promotes the growth of something we call sooty mold. And then there's the leaf shelters, the cocoons, webbing, as well as galleries and sawdust and pitch. So what I'm going to do now is we're going to talk a little bit about the injury that you're going to see. We'll go through a nice gallery, but bear in mind that most of the injury to crops is via feeding, whether it's chewing, like a chewing biting type injury, or a piercing sucking, or a rasping injury. So I have these little icons that I put here. And when we're going through the gallery together, you can see that little symbol just as a reminder to the type of mouth parts that you're actually looking at. So this picture here is of a peach, and it's fairly early in the season, and it was injured by an insect with piercing sucking mouth parts. In this case, it was oak bug, which is not something that we commonly find here. But you'll have other insects that can cause this kind of injury, like the tarnished plant bug, which is very, very common here. So you can see the little pinpricks there, and you can see this kind of ooze that's coming out of it. It's very sticky. We refer to this as gummosis. So if you're out scouting for that, this is actually one of the signs that you're looking for early in the season for insects with these piercing sucking mouth parts. The larger holes are actually the result of other types of injury, or their older injury, old stuff like that. So these are actually, this apple is actually injured by a stink bug. So stink bugs can be a real problem in orchard crops as well as other systems. This injury occurred late in the season in apple. So the stink bugs have these piercing, sucking mouth parts as well, kind of like a straw. And if you look very closely to the center of the holes in this picture, you can see the little pinprick points of entry where the mouth parts were inserted into the fruit. So it takes time for that darkening to kind of occur and that depression to occur. But you can find it when it's sort of a little bit earlier. Like I could have probably recognized this a few days before the point where this photo was taken, but it's certainly really obvious by this point. So this is an injury in apples, stink bugs in tomatoes. The injury looks more like a halo, and when you cut that open, the flesh underneath is kind of a corky white color. So this is kind of a neat thing here too, because you see these funny little bumps on the leaves of these grapes. And what that actually is this is these little insects are called grape phylloxera. Phylloxera is an indirect pest of grapes. It damages the vines by feeding on the roots, the leaves, but it doesn't actually feed on the fruit. But what happens is when a lot of leaves are injured, that really decreases the capacity of the plant to photosynthesize, and that can affect the grape quality at the end of the season. But it does take an awful lot of injury to do this, just because grapes have a lot of extra foliage. These little phylloxera are kind of aphid-like. So they're feeding within the plant. What happens is they start feeding on new green tissue, and this kind of little gall forms, and they feed within the gall. So if you were to cut open the gall, you would find the female in there with her eggs, and eventually those eggs will hatch, and the new little crawlers will move out, and the female, she dies in there. Very sad. If you were walking into the field one day and you found patches or rows of small plants that had fallen over, and then on close inspection you find that they've been clipped, your culprit may well be cutworms. So cutworms are the larvae of the noctuid moths. They're kind of not very attractive, kind of greasy looking things. And they like to hide during the day. So if you need to kind of confirm what your suspicion is, if this is cutworm injury, you need to dig into the soil to find your culprit there. So these leaves that are here have a lacy appearance. All the tasty green tissue between the veins has been chewed away. And there's a number of different insects that kind of cause this type of injury, which we refer to as skeletonized leaves. In this particular case, this is Japanese beetle that had caused that feeding injury. It is an invasive pest. It's been here for quite some time, but it feeds on lots of different crops. And actually, the larval stage is also injurious, but it doesn't feed above ground. It feeds in the soil on the roots of plants. But it is quite a significant pest. If you were to walk into a field and see small shiny insects jumping off the plant as you approached, and you see shot hole damage on those plants, chances are you're looking at flea beetles. There's many different species that are out there. Some of them have kind of a wide range, some of them have more of a narrow range. But these insects can cause a lot of aesthetic injury to ornamentals and to leafy greens. Most crops suffer yield impacts when they're really, really small, but they can grow out of the injury unless the numbers are really high, which they can be in some years. So if you wonder about why they're called flea beetles, it's because they can really, really jump. They've got these amazing hind legs that really allows them to propel themselves quite far away, and they will definitely jump when they see you coming. So what's kind of neat with this picture here is this actually looks to me a lot like a disease. This is a cucumber leaf with these black flecks on it. It looks a bit like angular leaf spot, but actually it's injury caused by the four-line plant bug, which is another insect with piercing, sucking mouth parts. The injury can show up really, really quickly because the insect inserts a lot of this kind of toxic saliva into the plant that helps break down the tissues. And actually, this is how the injury manifests itself after a little bit of time. Now, what you're looking at there is you see an immature four-line plant bug, and it is actually feeding on lavender. So these pear leaves have really ugly blisters on them. The undersides have this kind of nasty black spots. You probably realize it isn't a disease, But where are the insects that are causing this injury? Well, actually they're tiny, tiny little mites. They're pear blister mites that can only be seen under magnification. These mites look a lot like a carrot. They have two pairs of legs at kind of the fat end. And it does take really heavy infestations to cause a lot of injury, but this particular I guess tree had an awful lot of injury to it and probably would have had an impact on yield or fruit quality. So these grape leaves have a lot of stippling on them. One of the things you might have noticed while you were walking through the vineyard to actually take a look at this leaf and find, or these leaves and find out what's going on, is that a whole bunch of little insects, again, jumping ship as you're kind of walking through. If you carefully turn the leaves over, what you might find underneath are leaf hoppers. Now, typically the adults will fly away, but you may find some of the nymphs that are present. These are actually all adults, and there's several different species of nymphs, of leaf hoppers that do feed on grapes and cause this stippling injury. They're a few millimeters in size, so they're quite small. So it's really a numbers game. It took quite a lot of leaf hoppers to actually cause this injury on grape. So in contrast, though, if you look at the picture of the grape leaf on the left, it doesn't have that stippling to it. It's starting to curl under at the leaves, and there's some pictures of hops as well, where the leaves are starting to get this kind of marginal necrosis. And what's happening with this here is that there's an insect called the potato leaf hopper, which doesn't overwinter here. And when it feeds on plants, it doesn't take very many of them to actually cause this level of injury. It's just really, really toxic saliva, and it causes this kind of major change that occurs in a leaf. So the thresholds are very, very low for this one, in contrast when you compare it to those other leaf hopper species. So the injury in these pictures is actually caused by spider mites. They also have those piercing, sucking mouth parts, so just like those leaf hoppers. So it looks kind of similar, doesn't it? It looks a lot like leaf hopper injury. Well, so you're seeing some discoloration. You might see some webbing. If you look with magnification, you might see spider mites and their eggs. And I really love the picture that's on the top left here because it shows a spider mite next to a grain of salt. And I think it's really a kind of a brilliant contrast here. But these can be very, very serious pests in lots of different agricultural crops. They cause aesthetic injury as well as losses to crop yield. And they are really prone to resistance. They love really hot, dry conditions. These terminal leaves on peaches look absolutely terrible. They're all curled and deformed. If you turn them over, you will find colonies underneath, because aphids do feed in colonies. In this case, they're green peach aphids. So these insects also have piercing, sucking mouth parts. And when you find aphids, you'll often find predators that are feeding on them. They must be very, very tasty. These are some aphid midges in the top left-hand corner that are actually actively feeding within that colony. But again, you will also find things like ants that are tending the aphids and protecting them. And in exchange, what they're doing is they're feeding on the honeydew. And I have a picture of that in just a moment. And one of the things with aphids that helps to identify them and compare them to something like, let's say, a leaf hopper, are these cornicles or tailpipes that you find on the back end of it. So I mentioned this honeydew. So there's a great big drop of honeydew that is coming out of the back end of this aphid. And you can see there's an ant in another picture that's quite happy to consume that. Now, if that honeydew is present in large amounts, it can really cover the kind of the understory of whatever it is that the aphids are feeding on. And then you can get a buildup of this sooty mold as well, which is shown on the apples in that picture here. These blotches each contain a sawfly larva. The female lays her eggs between the upper and lower leaf surface, and the larvae feeds on the parenchyma in between. The feeding chambers become bigger over time. You can see the larvae in its frass when the blotches are cut open. Eventually, these would pupate and the new adult would emerge from the leaf. There's many different insect orders that actually mine leaves, and some of them, again, are very serious agricultural pests. And then the last insect that we're going to look at in our gallery are these thrips. So these are onion thrips, and the discoloration that you can see on those leaves is caused by thrips feeding, and probably from egg laying as well, because that does also cause injury on the plant. So these insects have that kind of a scraping mouth part they scrape the surface of the plant and they kind of suck up the contents from the liquid that is exuded. Now these plants here are way past threshold levels. The thresholds for thrips are usually quite low. It's really important to get your ID right for the thrips as well as with many of the other pests, because they may look similar to one another superficially, but you can have different species and their response to different pesticides can be really variable. I just wanted to finish off with a few additional slides on scouting for insects and mites. One of the things that I think you really need to do as scouts is to be aware of what the different pests are in the crops that you're working with. So make some kind of a map of when you would expect to see those pests and what the pests actually are, and learn to recognize them. You need to be sampling weekly, at least. In some cases, for some pest situations, things can change very quickly. You might need to be out more often. It is important to come up with a plan for how you're going to walk through the blocks that you're working in. So you want to make sure that you get a good representation of what's actually out there, whether that's a W or a Z. Those are some of the patterns that people talk about, plus borders and hotspots. You need to map your route weekly. Don't use the same route like every single time. And you really do need to be using crop-specific sampling protocols related to the number of locations in a field or the number of samples per location, the life stages that are present, the percent leaf damage, whether there's beneficials and other things. You need to write everything down because those zeros are actually really, really important. Don't be afraid to pull plants apart. You might need to do that depending on the crops that you're working in. One of the things that I found was a really good tip is the whole using kind of the position of the light to help you with your sampling. I mean, you don't want to be looking straight at the, like, it's hard to look great at a plant if there's the sun shining in your eyes. But sometimes if you're walking under a plant that has like a canopy, like an orchard situation, or in this case, corn, you can use the sunlight dappling through the foliage to actually see certain pests. So in this case here, this egg mass was showing up on the other side of the plant and the sun was shining through and it was evident. It's a picture by my, my colleague, Tracy Bowdy, who works in field crops. There's that. And then just a final word on toolkits. I have my own preferred tools. There's kind of the standard kits, that all the different things that you have, that I love to have tweezers and forceps with me, and paintbrushes are incredibly useful. I use what are called pooters, which are kind of like an insect aspirator. It's something you use to suck them up into a container so you can look at them. I like to have a nice saws and multi-tools, trowels and clippers. I like to have popsicle sticks actually for removing insects from pheromone traps. I like to keep paper clips and bobby pins that can help really secure my liners to the traps. Clips, garden twine, duct tape, believe me, it has many uses. Flagging tape and lots of it, different colors, but good colors like bright ones like orange or pink are excellent. Yellow and green, not so much. Tapping trays are useful, sweep nets. Containers for insect samples, I like to have bags, both paper and plastic, as well as lots of extra trap liners and lures and posts. Sharpies, I lose a lot of them. I try to put flagging tape on them so I don't lose them, but I still like to have lots of them. And of course, other writing implements, gloves, wipes, and I like to have essential oil with me too, just to remove the sticky goo that I get on my hands. So that's it. That's this presentation, basic intro to entomology, and I look forward to meeting up with you in some of our the subsequent presentations that are recorded on this website. Thanks very much for coming. If you do need to reach me, the best way is usually by email. My address is hannah.fraser@ontario.ca. You can always try me on my cell phone as well, 905-708-8014, but that changes. If you want more details about some of the things we talked about today and you're super interested, there's some fantastic resources that have been put out by Dr. Stephen Marshall at the University of Guelph, who's put out books on flies and beetles, as well as sort of the general insects of North America. These are beautifully produced books, lots of great pictures and some excellent information. Thanks very much. I look forward to speaking or seeing you again. Bye. Hi everyone, this is Hannah Fraser, entomologist for horticulture crops with OMAFRA, and this presentation is on making insect traps work. Traps are excellent tools for monitoring pest activity. They can be standalone or used to supplement other techniques. The information obtained is used to determine presence or absence, surveillance, pest activity patterns, or to initiate biofix. Information on pest pressure, or determine if action thresholds have been reached. In some cases, traps can be used to trap out or mass trap insects to help reduce numbers in the field or in the greenhouse. Many traps take advantage of insect behavior, including shelter seeking, movement through an area, attraction to certain colors or patterns or shapes, or directed movement to attractive volatile scents, including attraction to food sources or to pheromones. Pest management programs most often use compounds that attract to mate, i.e. sex pheromones, or call to others to a suitable food or nesting site, aggregation pheromones. You can think about them as unique perfume blends that are specific to a given species. Physical traps intercept crawling or flying pests as they travel. Double-sided tape is used to intercept crawling or climbing insects, such as the crawler or mobile stage of scale insects, as they move along trunks or branches. Corrugated cardboard is used here in this example to monitor or capture a codling moth, who like to hide in bark crevices to overwinter or to pupate. The barrier wall and pitfall trap shown above was used to direct wandering strawberry root weevil adults to trap them out and to discourage movement into uninfested plantings. Weevils also love to hide during the day, so physical traps like these blocks of wood are also useful for monitoring. Some colors are more attractive than others to both target and non-target insects alike. White often catches more non-targets, but it is the most commonly used traps that are commercially available. Work completed in the greenhouse by my colleague Sarah Jandrisic showed that plain yellow cards caught more thrips than their blue counterparts. On pests like whiteflies, fungus gnats, shoreflies, and leaf miners, there was also much more attraction to yellow, which makes it a better fit for comprehensive IPM programs. Trap design is also important because the size and structure of the opening affect the way the air moves through the trap and ultimately the pheromone plume emitted. Work by Rebecca Hallett at the University of Guelph demonstrated that Jackson-style traps, white ones, were far more effective than others for evaluating and monitoring the swede midge, which is a very serious pest of brassica crops. One of the things that you will find when working with sticky cards is that they attract a lot of different insects in all sorts of debris. You need to be able to locate and correctly identify your target pest on these traps. Sticky panel traps like these are the reason I keep essential oils with me while traveling to the field. Once my traps are all set up, I use them to remove the last of the glue from my fingers. It really, really works like a charm. My passion for using a hand lens has really decreased with my deteriorating eyesight over the years, but I found my phone to be an amazing substitute for zooming in on small details. Even without any extra gadgets, I can zoom in to identify spotted-winged Drosophila males on these cards. Given these insects are less than 2 millimeters in length, that's pretty impressive. On the left, there's some pictures of European cherry fruit fly, which is an invasive pest that's made its way to Ontario, and I can actually see some of the unique features on its wings that can help me to identify this two species. It probably isn't surprising that we can lure insects with scents that mimic potential food sources. We used apple cider vinegar for years to monitor for the spotted-winged Drosophila before commercial lures that were more selective were developed. Wireworm population densities can be assessed by placing food baits in the soil. Ambrosia beetles can be monitored using ethanol, which mimics what a stress tree emits, something beetles are attracted to. And Japanese beetle traps incorporate a floral lure and sex pheromones to trap both males and females. I wanted to mention a few things about pheromones specifically because we do use them so often in agriculture, particularly the sex pheromones. But these are chemical messages that trigger a behavioral response in other members of the same species. And again, they can be aggregation pheromones, sex pheromones, alarm pheromones, food trails, and others. Aggregation pheromones are usually attractive to both sexes, and they tend to be common among insects that exploit food resources that are kind of patchy in distribution or sporadically available. Sex pheromones are used for attracting mates. Males can produce them, but they are typically produced by the females to attract the males. The advantages of these sex pheromones is that they're very, very sensitive. They capture pest insects that are present at very low densities or that are hard to detect through scouting activities. In addition, because they capture only one or sometimes very closely related species that might have a similar blend, this makes identification and counting very much easier. So they're extremely valuable tools. It's important to use them. Pheromone traps are extremely useful tools. We can use them in a number of ways in agriculture. One of them is to determine the biofix for initiating degree day models. Now, I do have another presentation where I discuss the importance of temperature in determining the rate of insect development. We know using certain developmental parameters how long it will take for an insect to reach a given life stage. So if the time to hatch or the time till you see adult activity. And this information can be really important in terms of helping to time some pesticide sprays. Pheromone traps can also be used to help determine economic or action thresholds. For example, the number of insects in a trap per day. This is the point at which pest control actions would be taken to avoid economic losses. And of course, it's going to vary by insect. One of the things that's really important with this insects per trap per day thresholds is it is based on using a certain type of pheromone lure as well as a certain type of trap. If you're not using a standard, this can change things. One of the things as well is that the accuracy of the data is key. So make sure that you're recording any zeros in your pheromone traps. This information is incredibly important. And again, stress that you need to make sure that you're using the right trap for the job at hand. Insects respond to pheromone blends that are specific to their species. Commercial lures approximate these blends. Sometimes the blends of closely related species are similar, but they may have different ratios of the individual chemical components, or they may have minor compounds that others do not. What is really important is that you need to make sure you don't cross-contaminate your lures. Wear gloves when you're handling them, and make sure if you're working with multiple lures that you change them frequently. This same applies to the use of forceps or any other equipment that is used to handle lures. Make sure to take old lures and lure packaging out of the field. The lures need to be kept cool in transit to the field. Don't let them heat up in the extreme temperatures in your car, and make sure to store any unused lures in the fridge or freezer as directed by the manufacturer. Some traps can be used over multiple years. This is okay, but the traps need to be labeled so they're set for the same species every year. This is because the plastic in the traps can become contaminated. One of the most common lures is a red rubber septum, which resembles a pencil tip eraser, although the one shown here is white and others are gray. The septum is preloaded with pheromone. Field placement of traps is really important as well. If you don't put the traps in an ideal location, you may be missing out on some of the most important or accurate data. So this might mean a specific position within the crop, such as putting them at borders or even outside of the crop. It's very important to get the height right as well as the orientation. And make sure to use the recommended number of traps per area. And of course, this will vary depending on the crop and the target pest. You don't want to have your traps too close to one another because they may interfere with one another. It's a good rule of thumb to use at least 40 meters between your traps, and sometimes this needs to be much larger. Traps should go out 1 to 2 weeks before the expected activity of the target pest. This is particularly important when you're trying to determine a biofix because you do want to have some zeros before you start finding these insects in the traps. Mark the location of the traps well in the field. Use lots of flagging tape or flags and make some maps. This is just, when you think about it, this apple orchard where these traps are located in these pictures, it's, there's not much going on out there. There's not much foliage, but in the next few weeks, this will all fill in. And if you haven't marked the location of your traps, you're going to have a hard time finding them. And make sure the traps don't interfere with the normal grower practices. This slide just really illustrates the importance of height. There was some experiments done several decades ago with codling moth, and what the researchers determined was that there was definitely an optimal trap placement for capturing the maximum number of codling moth males. And this was actually in the upper 2/3 towards the top, but not at the top of the canopy of the crop. So make sure you're putting your traps in the right locations in the field. Maintaining traps is also critical. The data that you get out of them is really related to how well you manage your traps. One of the things that I do notice is that a lot of people just throw the lures into the sticky bottom traps that they're using, and that can be okay, but the problem is that those lures can get glue all over them, so you need to make sure that that really doesn't happen. I also like to mark my traps so that I put down our organization name, I put down the target pest name, and I write the date that the lure was and trap were actually set up. Just some reminders for myself for when I actually need to change the lures as well, because they do need to be changed. They don't last, many of them don't last the entire season. There's a few that do, but most of them do have to be replaced, and that time varies. So again, you need to make sure you're replacing them when you need to. And you do need to be checking your traps regularly. If possible, I like to remove the target insects and then I record the data. And oftentimes, if you have a pest that is abundant in very, very large numbers, you might even have to replace those liners or traps every week as well. This is a great little trick for installing the lure into the traps without it having to stick right into the glue. You can use a pin or a paper clip to push it through the top part of the trap. This works well with plastic delta traps that have some rigidity to them. And then you push the little rubber septum onto the end, and then it hangs down into the trap, and it doesn't get sticky. It doesn't get lots of debris on them. And this makes a lot of sense, especially when you're changing those liners frequently. Traps don't last forever. I do occasionally come across fields with traps that should have been replaced long ago. For example, the trap on the left, that is a diamond trap, and it's a sticky trap. And they're wonderful traps, usually very economical, and so they're heavily used. But the problem with them is that when they get really, really wet, over and over again, they start to lose their shape, and they start to just kind of pull down so that there's almost no opening to them. When the traps are like that, they're really not very useful. They need to be replaced long before that. The trap in the middle, that is apple leaf curling midge, and that picture would have been taken probably last week by my colleague. And this is what happens with apple leaf curling midge in heavily infested blocks. These traps fill up very quickly. And so those traps liners need to be replaced every single week. And then I came across this other trap, which actually has a camera in it to try to identify the pests as they come flying in. It's kind of a neat thing. But the problem is that liner is absolutely useless at this point because there's just too much going on there. So that should have been replaced earlier. And sometimes you find surprises in your traps. The liner on the right has a codling moth, and then it has another species that looks a lot like codling moth, but it isn't. It is another tortricid, and it just happened to end up in the trap. Maybe there's a component of the pheromone blend that fooled it, and the male flew into the trap to see if there was a female there. It does happen. So off-catch does happen. You need to be able to recognize your pest. And then unfortunately, if you look at these bucket traps on the left, what you'll see is that I managed to trap out lots of bumblebees at one location. I was very upset about that. And then I also had some mice that were nesting in some of these traps, which was not a very pleasant surprise to find at the end of the season. If you want more information, please feel free to send me an email. That's really the best way to reach me. And my colleague, Christy Grigg McGuffin, has put together a fantastic video on trapping, tapping, and visual observations in apples. And so I recommend that you check that one out. It'll give you some really good tips, even if you're not working in that crop. Thanks very much for your attention. Have a great day.
My computer is designed to be really slow right now. All right, can everyone see the presentation now in presentation mode?
Yep, looks good.
Okay, so I'll try and get us on track. Hopefully we don't go over too much. And I'm sure everyone's been waiting to hear this presentation, pesticide safety. It's usually a favorite. So to start off, I want to get you thinking about pesticide safety. So I have 4 pictures of different crops here. So pesticides may be applied to various crops. So before you enter any of these fields, which ones do you have to consider potential pesticide exposure? So I'm not going to run a poll, I just want you thinking about it. So A, corn, B, raspberries, C,
Amy. Thanks for pitching the lunch.
D, organic crops, E, A and B, corn and raspberries, or F, all of the above. So it is F. Most crops are sprayed with a pesticide at some point during the growing season. So it's good for you to know that. And when we say pesticides, we do include biopesticides or pesticides that have a natural substance, I guess you could say, in them. And so there are conventional and organic pesticides, and another term that we use for pesticides is pest control products. So you're probably familiar with Roundup herbicide, which contains glyphosate and is a conventional pesticide. Some examples of organic pesticides are dipel, which includes BT, which is Bacillus thuringiensis, and Pure Spray Green Spray, which is made up mostly of mineral oil. There are also pest control products that contain pheromones and are used for mating disruption. And as I mentioned, most crops are sprayed with a pesticide at some point during the growing season. So when you're going out to scout a field or an orchard, err on the side of caution and consider the possibilities that the crop you're entering may have been sprayed. Because you do not want to enter a field until you've confirmed whether it has been sprayed, what it has been sprayed with, and when it was sprayed, because you need to be able to protect yourself. So just to give you a bit of background on pesticide use in Canada, all products that are applied for pest control must be reviewed and registered by Health Canada's Pest Management Regulatory Agency, so the PMRA. We really like to use acronyms in agriculture. So before a pesticide is registered in Canada, Health Canada scientists must first review many scientific studies to ensure that the pesticide can be used safely according to the label directions. So these studies are on the efficacy of the product, residue studies, studies of the plant metabolism, soil dissipation, occupational exposure, studies on how it affects non-target organisms, all those types of things. And so they do assessments on that. And so the product label contains the label directions, and this is based on the data that the PMRA has reviewed. And the product label outlines how the product can be legally used. And it specifies safety protocols to minimize worker exposure during and after applications. So you can be exposed to pesticides if you enter a treated area too soon after an application. And exposure can be oral, so ingested, dermal through the skin, and respiratory, so through inhalation. So over time, pesticide residues and vapors dissipate until they no longer pose a risk to workers entering the fields. And the length of time required for these residues and vapors to dissipate varies with the product, the rate used, and the type of work that you need to do in the field, and also the environmental conditions. So how do you know it's safe to go into the field, the orchard, vineyard, greenhouse, whatever crop you're scouting? So through PMRA's evaluation of how the pesticide will be used, they come up with a restricted entry interval, which is the period of time that agriculture workers or anyone else must not do hand labor in treated areas after a pesticide has been applied. And you will find these restricted entry intervals on the product label. And these REIs give time for the pesticide residues to break down to safe levels for work to be done. And so what exactly is hand labor? So hand labor tasks involve substantial worker contact with treated surfaces, such as the plants, the plant parts, or the soil. And I've provided a list of examples of hand labor tasks, which include things like planting, harvesting, thinning, weeding, and scouting is included in this. So because when you're scouting, you can be touching the plants to flip them over, like flip over the leaves to see if there's insects. On the other side, you could be brushing up against the crop when you're walking through the field. So it's important as a scout that you're aware of these REIs and you only scout after the REI has passed. And REIs can range from 0 hours up to several days, and they are specific to the product that was applied, the crop it was applied to, and the post-application tasks, such as scouting and harvesting, etc. And you also should be aware that REIs can change over time. So the PMRA will occasionally reevaluate a pesticide's registration, and they can change the REI. Also, the grower or sprayer applicator may apply more than one product at a time. So for these tank mixes, you always go with the longest REI. And sometimes you may not find a REI on the product label. So in that case, you should probably go with the 12-hour restricted entry interval. And as I mentioned earlier, you can find these REIs on the pesticide label. And you can find these Canadian pesticide labels using PMRA's pesticide label search app or their online tool. So you can search the internet for PMRA label, and this is what you'll see when you do this. And I just took 3 different screenshots here. My phone, when I So the first one is showing the PMRA's label search app. It's circled in red. And then I typed in, and so the second screenshot is what you see when you open up that app. And then I typed in the word copper because I wanted to pull up the, I want to search up the product copper 53W. So it pulled up a bunch of different products, but if I clicked on copper 53W, the second screenshot is what you will see. And you can click on or touch the approved label, and then it will pull up the label for you. And you can download it if you want. For the online tool, this is what it looks like. And you can search the initial criteria. So you can search the field and you can pull down, it's a pull down menu, and you can pick product name or the active ingredient if you happen to know that, or the registration number. And so quite often you're going to know the product name, and you have to be careful how it's spelled. Sometimes it's a little tricky there. It has to be spelled just right. And so you would type that in the criteria. And so I had selected a product name and put in Vasagran. And so it pulled up 5 different products. So in order to get the label, you need to click on the registration number in order to pull that up. Another place you can get these product labels is going to the agricultural chemical companies' websites because they'll have their product labels posted there as well. Also, the OMAFRA does have various crop protection guides that will include REI information. So for horticultural crops, we have a vegetable crop protection guide, and we also have 5 separate ones for the various fruit crops. So we typically update these annually. So as I mentioned, REIs can change over time if the PMRA has reevaluated it. So it's a good idea to make sure that you're always checking the most current label. And here's the guide to weed control for hort crops. So it's new one too, and there's REIs in that one as well. And I pulled up an example out of the apple crop protection guide. So this is a table of products used on apples that you can find in there. And they have a column restricted entry interval, and you can look up the REIs there. So if you look at the bottom for a sale, there's 3 different restricted entry intervals listed. So there's a 12 REI for general reentry, a 48-hour for contact and scouting activities, and a 6-day REI for hand thinning. So I mentioned before, they can be specific to the task. So that's important for you to know. And this is an example of our vegetable crop protection guide. So this is a brassica crop insect control. So in this pub, they include the REIs under the notes. So 12-hour REI here, 12-hour, 12-hour, and then you get to malathion. And you have a 2-day restricted entry interval for broccoli, Brussels sprouts, etc. But for kale and kohlrabi, you have a 24-hour REI interval. So it does matter which crops as well. So before you enter a crop, ask the grower or sprayer applicator, and possibly your supervisor will know this too, what was sprayed and when. You need to know this in order to look up the REI, because you don't want to enter the treated crop before the REI is ended, and it is safe to do so. Also, it's good practice that you let your supervisor know where you will be at during that day, and check with your supervisor on any products that may have some special instructions beyond the REI. There are some products that do have that. Also, if for some reason you haven't been able to get the spray records or application information, you can talk to your supervisor about that, because maybe they'll be able to help you out. And so to communicate the REIs, did I skip a slide? No, sorry. Ontario growers are using various ways to communicate REIs to scouts and other workers. So I'm not trying to promote any app over another one, but just want you to be aware that they are using different things. So some of the tomato growers are using Spray Hub to communicate what they've sprayed and when what the REIs are. Some growers were using WhatsApp. And now Signal's becoming pretty popular too, so some growers may be using that. Also, you can use text message, whatever works for you and the sites that you're going out, but you should develop some line of communication for that. It can happen though that when you arrive at the field, the crop has just been sprayed. And it's happened to me before. So miscommunication can occasionally happen. So it is important that you recognize the signs of spraying. So one potential sign of spraying is you can smell a distinct odor because some products do have that. Another sign of spraying can be a sign. So the grower can put up signs at the entry points to the crop so that growers will see that it's not safe to enter that crop yet. Another sign is that you might see tractor spray row tracks. If the tractor's just gone down, you'll still see those. Also, you may hear the sound of the sprayer nearby. So in that case, you should leave the field and you should talk to the grower or your supervisor because you might need to change your schedule for that day and come up with a plan B. That was the sound of the sprayer. Other signs of spraying is you might see residues on the leaves. So in this case, the foliage is still wet from an application. However, just because you see residue on the leaves doesn't necessarily mean that it's pesticide residue. So here's a picture of a whitish residue on leaves from calcium magnesium precipitates in irrigation water. So when in doubt, leave the field, call the grower, your supervisor, and to confirm what it is. And I hope no one encounters pesticide exposure during their scouting, but it's good for you to be informed on what you can look for in case this does happen. So potential symptoms of exposure can be headaches, fatigue, irritation of the skin, eyes, nose, or throat, loss of appetite, dizziness, nausea or vomiting, diarrhea, decreased muscle coordination, and blurred vision. So if you feel this way after you've been in the field and you think it could potentially be pesticide exposure, you should seek medical help immediately. Also, a good general practice would be to have a shower at the end of the day. And for pesticide poisonings, there is the Ontario Poison Center that you can call. I've provided the phone numbers for that center on this slide. And also you can always call 911. Again, I hope nothing like this happens while you're scouting this summer. I hope you have a great season. It should be a lot of fun. So that's it for me. So we're a little over time. Is there any questions on that?
Haven't seen any come in the chat.
Okay. Okay. So we're a little over time, but I'm hoping we can still come back at 1:05 from lunch.
Sounds good.
Okay, so we'll get started. Can everyone hear me okay and see the presentation?
Good.
Yep. All right. So for the next 20 minutes, I'll cover farm visit biosecurity. So what is farm visit biosecurity? So it's taking steps to reduce the risk of introducing and transferring plant pests within and between farm operations. And just so you know, there's no such thing as zero risk. You try your best to minimize it, but you can't guarantee that there is no risk. And so just how can pests spread? So there are natural means that you can't really do anything about. So certain pests can travel by wind. So spores from soybean rust is one example. Also insects can travel in the wind, and they can also fly. So flight is another means. Pests can also spread by human activity, so in international trade. So we can have, it can be done by plants and plant parts and seeds, grain. You can bring in pests that way. Also, some pests are excellent hitchhikers. So in the bottom picture here, there's 2 red circles around brown marmorated stink bug that came in on a shipment of PVC piping from an infested state. Another way that pests can move is on contaminated stuff. So such as vehicles, footwear, hair, equipment, clothing, and hands. So as a scout, these are the things that you should really be thinking about. You want to be able to protect your clients. You don't want to be a vector for a new pest being introduced to their farm. I like to show this study because I think it's really interesting. It was done by McNeil a few years back. And what they did was they surveyed organisms present in the soil that had been removed from footwear being carried in the baggage of international aircraft passengers that were arriving in New Zealand. And what they found is that they recorded high incidence counts and diversity of viable, so live bacteria, fungi, nematodes, and seeds, as well as several live arthropods. And in each gram of soil, there was an estimated 52 to 84% incident of genre that contains species regulated by New Zealand's National Plant Protection Organization. So that's a bit scary and surprising. So just to go along the footwear theme, so back in 2015, OMAFRA did a little soil biosecurity project. So we had the objective of this project was to determine the risk of footwear to obtain soil-borne pathogens, and we were specifically looking at nematodes and fungal pathogens when walking around in fields of 2 different soil types and under dry and wet soil conditions. And so we had 4 people walk around the field with clean rubber boots for 20 minutes, and we did the same fields under dry and under wet conditions. So we looked at a silt loam and a sandy loam, I think it was. And so we did collect the soil from each pair of boots using a hoof pick. And we also did take a background sample. So using a soil probe, we went about 8 inches down just to ensure what exactly was there. And so it was a good comparison to the soil that we collected from the boots. And so we sent all these samples into the University of Guelph, the Pest Diagnostic Clinic, and we had a DNA multi-scan run to look for fungal pathogens, and also they did nematode extractions. So what we found was there was a whole bunch of stuff that we found. A lot of this is common, like endemic pests, like Rhizoctonia silene is pretty common. The one field we knew there was a history of sugar beet cyst nematode, and also they have Plasmodiphora brassica, so that plant pathogen that causes clubroot. And those aren't found in every field. So that's why we did target that field, because we were interested to see if we could pick up those things. So what we found was the risk of transferring soil-borne pathogens on footwear was similar when walking around silt loam and loamy sand fields. And we did collect more soil and more soil-borne pathogens on footwear after walking around fields under wet conditions, which isn't totally surprising. For sugar beet cyst nematode cysts, we did detect on one person's footwear after walking around the silt loam fields under wet conditions. Also, root lesion nematodes were not detected on any footwear after walking around a loamy sand field, regardless of the soil conditions. And for the Plasmodiphora brassica, the causal agent in clubroot and brassica crops, it was only detected in the silt loam field, but silt loam soil, but we knew that field had a history of that. And the DNA of the brassica was detected on most footwear after walking around the field during both wet and dry conditions. And so in the picture you can see, like, sometimes we weren't getting a lot of soil on the boots. So it was a bit surprising. But just because we were picking up the DNA of these pathogens doesn't mean that it was viable. So we would have had to do a further study to prove that. But really what we wanted to do was demonstrate that there is some risk of transferring pathogens and nematodes on dirty boots. And Katie mentioned soybean cyst nematode earlier today. So this nematode can infect soybeans and can cause stunting and yellowing of the leaves, and it's frequently found in circular patterns in the field, and it can move in soil. So it was first detected in Ontario in 1988, and since then it is now found in most counties west of Toronto and is now found in certain parts of Eastern Ontario. So this disease can be managed effectively, but the first step is really identification and awareness. And so I did include a Farm Visit Biosecurity handout for you. So please refer to the revised one that I sent out this morning, because it's more tailored to scouts. The other one was more for our crop specialists, like when we're called out to diagnose the problem. So this one's a little more appropriate for you. And so this is a summary of the main points from our OMAFRA farm visit biosecurity protocol for visiting orchards, vineyards, crop fields, and greenhouses. And it shows the practices that you can do to help minimize, rather than eliminate the risk of disease, insect nematodes, or weed transmission under normal circumstances. And so there is additional biosecurity measures that may be required in situations where there may be a contagious plant disease or a regulated crop pest. So you might need to wear washable or disposable footwear and clothing. And but discussing concerns you may have with your advisor where farm visit biosecurity requirements may conflict with your safety. And so I'm thinking here, like if it's really hot out and wearing coveralls and that type of situation, you don't want to have heat exhaustion. So those types of things. And sometimes the plastic disposable booties can be pretty slippery. So depending on what the situation, that might be a concern as well. So I've just shown you a picture of what is in my biosecurity kit. So I do tend to carry a few pairs of rubber boots. I don't always have a chance, like if I'm trying to jam a lot in one day, to clean them after each farm. So I'll stick it in a plastic bag and clean it when I have time. I do also carry disposable booties, like the blue plastic ones, also the little hospital booties. So those I wear more for if I'm changing traps in the same farm and I want to try and keep down any soil down getting into the car when I'm hopping in and out. So I'll put those on and then I can take them off before I get back in and throw them in a baggie. I also use disposable gloves a lot. They're good to have if you're handling pheromone lures as well. Disinfectant wipes, always using them. Lots of bags, paper towels to wipe down equipment too. Kitty litter box that I use to clean my boots in, and so I always carry potable water as well and some detergent. The hoof pick to remove that soil before I do any cleaning, and also some disinfectants. So there are some brands of rubbing alcohol that are ethanol based, so I'll use those. There's also Burcon as well in tablet or powder that you can use. Some people use bleach. So it depends your circumstance and if you know the history of the field, what you're trying to protect from, and because disinfectants can be effective against different things. So before the visit, if possible, call ahead to the grower. And this is gives you a chance to see if there's any additional biosecurity measures in place on the farm. So in the case of like all of a sudden they are aware of a contagious plant disease or a regulated pest, you can have the heads up on that. And also just with the COVID situation right now, it gives you a chance to confirm that you can maintain that physical distancing and to see if there's anyone sick on the farm. So yeah, just follow precautions for the current COVID situation. It's best to schedule farm visits when to avoid walking in wet fields and touching wet plants as much as possible. Also, clean and disinfect any equipment that you're going to be using on the plants, and wear clean clothes and footwear. And upon arrival, it's best to drive slowly to avoid unnecessary contamination of the vehicle. Like you don't want to be zooming down the lane and then the dust is flying all over the place. Also, you want to roll up the windows. Yeah, this past year we had a gypsy moth infestation, so they were always trying to get in the vehicle. So it's always good to roll up your windows. Park in a designated visitor parking and or away from barns and other sources of contamination. So you don't want to park near air intake vents or ventilation and exhaust. Clean your hands with disinfectant hand gels. Ask to sign the visitor logbook. I don't know if things have changed with the COVID situation, that maybe the grower will have other practices in place. So maybe they'll want you to text instead, like when you're arriving. So it's best to check with your supervisor and the grower. And put on protective footwear and clothing before entering a field, orchard, vineyard, or greenhouse in high-risk situations. So again, like if there's a contagious plant disease or regulated pest present. and wear protective footwear and clothing if unable to assess whether a situation's high risk. So your advisor can probably help you out with that, and also the grower. So during the visit, if possible, leave the vehicle in a designated area and walk the crops. So the whole thing is you're trying to limit the exposure of your stuff to what's on that farm, but you can't always do that. So sometimes I'll have traps way at the back of the farm, and I have a lot of equipment to carry, and it's just not feasible that I'm going to carry all that. So just try and limit the driving through the fields as much as you possibly can, and drive slow. Obtain all signage and barriers, so you don't want to enter buildings unless permitted by the farm operator. And again, just follow any precautions for the current COVID situation. Also avoid unnecessary contact with the crop. So like certain diseases such as tobacco mosaic virus can easily spread by contact to other susceptible hosts. And wash and disinfect any equipment that will be used on plants between use on what we call biologically separate or unique areas and or buildings within the premise. And before leaving the operation, visually inspect your clothing, your hair, your equipment to ensure there's no insects or plant material present or attached. Ensure your footwear is free of excess soil and plant debris. Use a brush to remove that soil from the tread, and wash and disinfect your footwear. Wash and disinfect any equipment used during the visit, and wash hands or clean your hands with disinfectant hand gel. And cleaning your footwear. So as I mentioned, remove the debris and soil. You need to do that first because organic matter will bind to the disinfectant and it'll make it ineffective. So you're just wasting your time there. So it's really important to clean off that organic matter, rinse your footwear, dissolve its disinfectant and submerge it in your boots in that. So certain products do have an exposure time on the label. So for like Virkon, I believe it's like a 10-minute exposure. So make sure that you're following that. Sometimes people will spray their boots instead because they don't have time to soak it, so they spray it down really well. And before leaving the operation, leave disposable materials with the grower if you can. Another option is to put the stuff in a sealed plastic bag or washable sealable container so you can dispose of it later. And try and really keep clean and dirty material separate. And inspect the interior and exterior of your vehicle for any visible contamination. And that can be manure, insects, plant material, excess soil. And remove as much visible contamination as possible before leaving the operation. Contaminated vehicles must be washed. Well, this is our protocol before entering another agricultural operation. There are some additional measures if there is a contagious disease or a suspect new pest. So if you're taking a sample, place it in a container that's sealed properly so that you won't have anything get out. Visit the site with the known or suspected contagious or regulated plant pest last if you're doing more than one visit during that day. And boots and field equipment must be cleaned with a disinfectant agent before leaving that site if this contagious disease or regulated pest is soil borne. And wash the vehicle before entering another farm or facility. So the greenhouse growers, they're pretty strict with their biosecurity, and the Ontario Greenhouse Veg Growers and OMAFRA developed this postcard that growers can give to their visitors when they're coming to their facility. So it pretty much outlines a lot of the similar things that I just mentioned. And I already went over some of the stuff on things that you can carry. So these are the same type of things that I carry in my biosecurity kit. And I mentioned a few times during this presentation about regulated pests. So just so you're aware, the federal government does have an agency called the Canadian Food Inspection Agency, so the CFIA, and they have a plant protection program in place to help prevent the introduction and spread within Canada of plant pests of quarantine significance. So they have a list of regulated pests that they will take action on. And the actions that they take really depends on the pest. So, but it can mean that the crop needs, like it could mean that the crop needs to be destroyed if a regulated pest is found. It really depends. But I just want you to be aware that there is a responsibility to report new pests. You're supposed to report that to CFIA, but first you should check with your supervisor that it is actually a new pest. These situations can be very contentious and should be handled by your supervisor. So you should immediately inform your supervisor if you do suspect a new pest. And just showing an example of why it's important to check with your supervisor or someone that may have more experience than you, because some of you may be very new to scouting. So the big picture here is Asian longhorn beetle, and that is a regulated pest by the CFIA. And there are some lookalikes. So we do also have the white spotted Sawyer beetle down in the left corner, and that's present in Ontario and sometimes confused with Asian longhorn beetle. Also, some people do confuse it with the Western conifer seed bug. So that's why it's really important to make sure that you have the proper identification. And that's it for me. So is there any questions?
None have popped up in the chat.
Okay. Well, I'm here to the end, so if you do have any questions, feel free to ask. Okay, and next on board is Amber Holland, and she'll be talking about soil and scouting.
Come on. Okay, just checking to make sure that what you're seeing says soil and scouting for IPM.
Yep, it does.
Awesome. Because I'm never really sure. Okay, so we've been talking a lot about pests, but you have to remember the thing that we're growing in, unless you're scouting greenhouses, is soil. So it always comes back to the soil. And I love some of what Elizabeth was talking about at the beginning. She showed you some really nice pictures of soils and roots and things like that. So it's always important to not just assume that it's an insect or a disease or something like that. It could be something else that's going on in the soil, or that could be the vehicle for it too. So let's dive into this a little bit more. Let's see if I can get my, oh, for Pete's sakes, really? There we go. So the soil can tell you a story. And there's two ways of looking at it. There's diagnostics that you can do from field observation, and then there's diagnostics that can be done with lab samples. So some of the field things we can look at are looking at the soil profile. We'll look at that a little bit more. Texture, how well it's draining, how well water's going in, especially like say on a day like this where it's cool and nasty and we've been getting some rain. How well is that water moving? Do we have compaction? What's the structure look like? When we look at the diagnostic lab samples, there's a number that we can do. The basic fertility samples, looking at pH. pH is often one of the issues that we've got. Or could it be salts? And then of course, as Katie talked about, there's also nematodes. So in-field diagnostics, we've got a soil profile, and many of you have probably taken a soils course at some point. And we'll talk about like A horizon, B horizon. So the A horizon is the topsoil. And some of the things we can get from looking at the landscape is it rolling? Is it flat? A lot of our managed landscapes have been managed. They've been tilled and then they may have been leveled. So some of the variability you can see in a crop may be more to do with the depth of the topsoil. And a soil probe or even a shovel can tell a lot here. I can think of a few project fields where I started out thinking, this is a great project field, it's nice and level, it's consistent. But then when we got to harvest, because of course I was silly and I didn't look at it more closely at the time, I realized that our A horizon depth across the field was very different. They had at some point leveled the field. We can also have textural layers, and there can be some huge organic matter differences. The thing is, soil is not like water. It doesn't mix. It mixes a little bit with tillage, but we've got a lot of variability within any given field. So if we take a closer look at a soil profile, we can do this with a shovel, we can do it with a soil probe, or even an auger. And especially if you're into a situation where there's a lot more gravel or small stones, trying to put a soil probe in is frustrating to say the least, especially as the soil moisture goes down over the course of the summer and it gets drier. Anytime we've got small pebbles, it can be very frustrating. So a small Dutch auger is often a good option there. And you can see that we've got, if you look at the soil probe in the center, we've got a nice profile there where you can see the topsoil. The topsoil is about 6 inches deep. And you can see it grades into something that is more reddish. That's the B horizon, and that reddish color tells us that there's iron there. It means that there's been historically a fluctuating water table. And that tells you something about that soil in itself. So that soil probably somewhere below has a clay layer. And that tells you something about the whole moisture aspect in that field. The other thing we can tell with some of these in-field diagnostics is texture and structure. So texture is that sand, silt, and clay. You can either hand texture or do a lab test. We'll talk about hand texturing in a second. You can send it in for a particle size distribution test. Into most of the labs, they'll do that. It's usually about 40 bucks. It's fairly expensive. Those tests don't change though, and that will give you a nice breakdown on sand, silt, and clay. Structure, though, is how the soil is held together, and it reflects the density. It has an impact on water holding capacity, and of course, because it's water holding and density, it also will have an impact on how well is that crop rooted. Remember back to Elizabeth's pictures showing that the potatoes had stolons going out on the one, and the other side it was just kind of a crazy root system, and it didn't really know where it was going. So there's a number of things that can be shown by looking at the structure. Normally, in a normal year, I would be with you in person, and here's where we would get our hands really dirty and have a great time doing some hand texturing of soil. So it's not that hard, and this is exactly the way a soil pedologist, somebody who's mapping soils across the province, would do it. They take a small sample, they put it in their hand, they'd add a little bit of moisture, do the moist cast test where you've got a clump of soil and you basically bounce it in your hand and see how much it breaks apart, and then do a ribbon. So it's kind of that cigar, and then seeing if it'll flake as you push it across your finger. A field test to see if it's flowering. Once upon a time we used to do that by tucking it in the back of our teeth and seeing if there was grit. And then the shine test is with a piece of metal, a quarter, a knife, whatever you've got available. And of course, we start the whole process figuring out whether it's more or less than 50% sand. And this will bring us out to a rough idea of what texture is. And often that will help you just in answering some questions about what you're seeing. And if anybody wants that hand texturing chart, just let me know. I can email it to you. Texture is only part of it though, because when we start looking at structure, that's the piece that management creates out of that texture. It has a big impact on whether we've got erosion, we've got a compaction, how well those roots move through the soil. And so here's two examples. These are soils I'm really familiar with because they're actually at my place. And amazingly enough, they are the same soil type, they're the same texture, but when you look more closely, what are you seeing that's different here? And I'm going to ask you to put it in the chat, and maybe Katie can help me here because I can't see the chat right now. What are you seeing that's different between these two soils right off the bat? Okay, anybody awake?
Yeah, we got one answer of aggregation.
Aggregation is correct. That's one thing. What else do you see? And it's going to really depend on the quality of your screen, probably.
Crop trash is another answer.
Yeah, there's some crop trash. That's true. There is some differences here, of course, because the one on the left is coming out of hay with a lot of low tillage, but still a lot of traffic because it is hay. The other one's continuous corn with full tillage. In fact, these guys like to till in the dead of night. So we've got a difference in aggregation. The structure is much better on the hay part, and the water is going to go through a lot better. What else are we seeing? You don't see a color difference. I see a color difference. So the one on the left to me is darker, just a touch darker. And actually, if you saw these in person, the color difference is significant. The one on the left looks more like chocolate cake. And the one on the right actually looks kind of sad. Kind of like that soil again that Elizabeth showed us where the lime had been piled. So these two have a significant difference in organic matter, which plays out when we go to put both of them into corn and end up in the summer with a bit of a droughty situation. The one on the left is much more resilient. We don't see drought. We don't see a lot of stress on it, period. Whereas the one on the right, we're certainly going to see stress much more quickly on that one. So some of this is structure, some of it that's connected with structure is compaction. So we get compaction, it's caused by tillage and traffic under moist conditions. Doesn't have to be wet, and also equipment weight. We usually associate compaction with heavier soil types, but some of the worst compaction I've ever seen is on sandier soils. Because it will, if you think about how we build roads, soils that are poorly sorted, like a lot of our sands and sandy loams, if you think about how we build roads or how we build decks and lay down brick outside, we're usually looking for that kind of coarse sand that'll pack together tight, and we can do the same thing in an agricultural field. So that means that we end up with denser soils with restricted root system. Less pore space means less available water, so we get stressed, and anytime we stress a plant, it is opening it up to a lot of these other things that Katie and Hannah have been talking about. And the real kicker here is when you're walking a field, you're probably not going to see any of this because it has been hidden by tillage. So you need to be looking for patterns. You're looking for patterns with everything else, but these are patterns that often relate back to tractors and tractor spacings, harvester spacings, things like that. And what you'll see is we take it as common to see standing water on headlands, and that really shouldn't be. If you start digging up some roots and you see flattened roots, and I realize that's not the greatest photo that I've taken there with the corn, and sorry, I will work on that now that I've got Elizabeth's suggestions. Those flattened roots suggest that the roots are having a tough time getting through the soil, and we're more likely to see drought stress or restricted root systems. Like I said, we can see even slower growth. So if you look at the picture at the bottom, that's actually in a tomato field from Kent County. And a consultant asked me to come out and take a look at the field because the plants just weren't going anywhere. There was nothing wrong with them in terms of fertility. There was nothing wrong in terms of disease or insects. They were just sitting there going nowhere. And it all sorted itself out because we got a very timely rain. But what had happened was there was some compaction. You can see the compacted layer there. The roots really weren't getting out of it. And we had slowly been drying down. So those compacted soils, as they dry down, get even harder for roots to get through. And we had that platiness there that was really interfering with root expansion. So the other part of the diagnostics is soil sampling. And there's three basic kinds of soil samples: a basic fertility and pH sample that we take at the depth of 6 inches, and we can handle it at room temperature. Soil nitrates, something we don't take a lot of in horticultural crops, but sometimes you will, more with sweet corn or things like that. It's at a depth of 12 inches, and we need to keep it cool or freeze it. And probably one of the other most common ones is nematode samples. And we want to make sure we're taking those at the right time. We don't want to take them generally in the heat of the summer because soils tend to be hotter and drier. We're taking that at about 6 inches. It's a little bit deeper. Actually, it's about 8 inches. We're going to take the top 2 inches off, and we want to keep them cool and not freeze them. So when I go out soil sampling, my basic soil sampling kit means I have a soil probe. You can do it with a shovel, but it's really not recommended. It's really tough to take a good sample. You need a bucket that's clean, bags, boxes, a writing utensil, and depending on what of sample you're taking? Probably a cooler. And I like this one because I can drag it through the field pretty easily. And we're looking at diagnostic samples where, just like with your insects and diseases, we're going to look at the good areas and the bad, and sometimes we'll even look on the fringe just to see what's changing in terms of pH or fertility. Now, nematode samples, like I said, are a little bit different. We're going to take them so that the core that we end up with is 6 inches, but we're going to take, and I'm mixing my units here, I apologize for that, I can be bilingual. We're going to take about 7 to 8 inches and just take the top couple inches off. And the reason we do that is because when we're doing a nematode test as opposed to the DNA analysis, we're depending on those nematodes being alive and being able to swim out of the soil sample. So they have to be alive, and that top couple of inches is really prone to a lot of diurnal flux. So you get a lot of change in moisture and temperature, and it tends not to support a big population of nematodes compared to below ground. And of course, we want to handle them gently and keep them cool, not cold, not a big shock, and get them to lab as soon as possible, because again, the nematodes need to be alive. And that's it, unless there's any questions.
Not seeing any questions.
Well, thank you. Sorry we couldn't do this actually out in the field like normal. Maybe next year.
Yeah, thanks, Anne. Usually people love to do that exercise with the soil and get their hands dirty. So now we have Tajendra, who will talk about nutrient deficiencies and tissue sampling. So are you going to try and go to your presentation? Okay, awesome.
Good afternoon, everybody, and thank you for joining us today. First of all, thanks to Denise for including me to the IPM intro group this year. And I'm really excited to be here talking about deficiency symptoms and tissue sampling procedures in horticultural crops. And today, instead of going live, I would like to play a video of my presentation, which was recorded early this week. And I just wanted to see if the Zoom recording could be used as an alternative to live presentation. So please feel free to write your comments or feedbacks on the chat box. I will be here until the end of my presentation to respond to any questions or comments you have. And please bear with me for a second until I share my screen. Hello everybody, my name is Tejendra Chapagai. I'm your new soil fertility specialist in horticultural crops with OMAFRA, and I'm based in Guelph. Today I'm going to talk about major symptoms of nutrient deficiencies in horticultural crops. I would also discuss tissue sampling procedures which help to correct deficiency or toxicity of nutrients in crop plants. Before jumping into the deficiency symptoms, I would like to talk a little bit about essential nutrients for crop plants. And nutrients are mainly divided into 3 different categories based on the quantities required for plant growth and reproduction. They are primary nutrients, secondary nutrients, and micronutrients. And primary nutrients are required by the plants in the large amount, while secondary nutrients are required in the moderate amount, and micronutrients are required by the plants in the small amount. And primary nutrients are also called as the macronutrients. And there are 6 nutrients that fall into this category, and they are nitrogen, phosphorus, potassium, carbon, hydrogen, and oxygen. And plants acquire carbon, hydrogen, and oxygen from the air and water. However, the remaining macronutrients must be obtained from the soil. And there are 3 nutrients that fall into the category of secondary nutrients. They are calcium, magnesium, and sulfur. And referring to the micronutrients, they are as important as the primary and secondary nutrients, but they are needed in much smaller quantities by the plants. And there are 7 nutrients that fall into this category. They are boron, zinc, molybdenum, manganese, iron, copper, and chlorine. So in this presentation today, we are not talking about the roles of each nutrient. Instead, we are talking about some major symptoms when these nutrients are deficient in plants. And it is important to note that these symptoms can be confusing because many plant nutrient deficiencies share the same or very similar symptoms. Also, they can be similar to symptoms of many plant disease. For example, the symptoms due to deficiency of nutrients can be observed in different plant parts. And when we observe some symptoms first in younger leaves or growing points, that could be because of deficiency of sulfur, iron, manganese, or copper. This is because plants do not mobilize these nutrients from older to younger leaves. But when the symptoms are first visible in older leaves, that could be because of the deficiency of nitrogen, phosphorus, potassium, magnesium, zinc, or molybdenum. And it is important to note that calcium and boron deficiency symptoms seldom appear on older leaves. The other example is leaf chlorosis. And when this symptom is combined with retarded or stunted growth of plants, that could be because of the deficiency of nitrogen, sulfur, or copper. But if the growth is not retarded, that could be associated with phosphorus deficiency. Similarly, when nitrogen is deficient in plant, we observe yellowing of older leaves. But when the plant is deficient in sulfur, we observe yellowing of younger leaves. That means symptoms can be confusing in many cases. And this image also shows that the deficiency of boron, calcium, sulfur, iron, manganese, and copper are first visible on younger leaves, while the deficiency symptoms of nitrogen, phosphorus, potassium, magnesium, molybdenum, and zinc are first observed in older leaves. Now, let us talk about the major deficiency symptoms of nitrogen, phosphorus, and potassium. And when the plant is deficient in nitrogen, the most common symptoms that we observe are yellowing of older leaves, thinner stem, and stunted growth of the plant. Because nitrogen is mainly responsible for greenness and leafy growth in plant, and it is the part of chlorophyll and plays crucial role in photosynthesis. And the typical symptoms associated with phosphorus deficiency in plant is reddish or purplish lower leaves. We can also observe poor flowering and fruiting as well as premature fruit drop due to phosphorus deficiency. And when the plant is deficient in potassium, the tips and margin of the leaves turn yellow, and the plant becomes vulnerable or susceptible to stress conditions, both biotic as well as abiotic stress conditions. So the difference between nitrogen and potassium deficiency symptom is that if the plant is deficient in nitrogen, the yellowing starts at the tip of the leaves and proceeds towards the base. But if the plant is deficient in potassium, the margin of leaves shows scorched or burned symptoms along the edges. And the major symptoms associated with calcium deficiency in fruits and vegetables involve rotting from tip of the fruits, which we refer to as blossom end rot of fruits and vegetables. And we can also observe dieback of young leaves and buds, which we call tip burn. We can also observe bitter pits in apples and black heart in celery due to calcium deficiency. And when the plant is deficient in magnesium, we usually observe yellow and white patches between green veins of leaves. Plants also produce poor flower and low quality fruits due to magnesium deficiency. And symptoms associated with sulfur deficiency are observed in younger leaves. This is because plants do not mobilize sulfur from older to younger leaves. So younger leaves turn pale green to yellow, And we also observe stunted growth or retarded growth of plants due to sulfur deficiency. And when the plant is deficient in boron, we may observe hollow stem in brassicas. We can also observe internal cork of apples or deformed apples with brown spots. We can also observe cracked stem in celery. We can also observe hot rot and girdle of beets and dieback of plants starting from the terminal buds due to boron deficiency. And the common symptoms associated with zinc deficiency are development of small leaves, which we refer to as little leaf symptoms. We also observe yellowing of young leaves between the ribs, wilting, poor fruiting, and dieback symptoms due to zinc deficiency. And when the plant is deficient in molybdenum, leaves turn yellow and pale between veins. In addition to that, leaves may become bluish-green or they may not open completely. And the common symptoms in cauliflower due to molybdenum deficiency is whiptail as seen in this image. Similarly, when plants are deficient in manganese, we may observe brown spots on leaves along with stunted growth or delayed maturity in plants. While intraveinal chlorosis in young leaves and stunted growth are major symptoms associated with iron deficiency. And the common symptoms associated with copper deficiency are wilting of young leaves without yellowing and leaves developing brown spots. Similarly, we can also observe dieback from leaf tips, development of multiple buds, and production of gum pockets due to copper deficiency. And in general, chlorine deficiency is not common in fruits and vegetables. However, if this nutrient is deficient, we may observe wilting of leaves, especially at the margins. We can also observe curling of leaves, bronzing, chlorosis, and necrosis. Now, I am going to talk about the second component of my presentation, which is tissue sampling procedures in horticultural crops. And tissue sampling is important to identify if a specific nutrient is excessive, adequate, or deficient during the growing season, and to correct immediately if they are deficient or if they are more than adequate in crop plants. Also, tissue sampling and analysis is important because there are no accredited soil tests available for boron, copper, iron, and molybdenum. And plant tissue analysis can provide information to help manage these nutrients. But it is important to note that tissue analysis could be unreliable sometimes for evaluating nitrogen and zinc due to timing and growth stage of the plant. Therefore, plant tissue analysis is most useful when it is combined with the soil test, along with a visual inspection of the crop and soil conditions. The table here shows sampling time as well as in some information about what and from where to sample. This also tells some precautions that need to be considered while collecting tissue samples from tree fruits and grapevines based on OMAFRA publication 611. And as we can see in this table, the sampling time for tree fruits is from late July to early August, while the sampling time for grapevines is from early to mid-September. In general, we do collect at least 100 leaves for tree fruits, while we collect at least 100 petioles from grapevine for each sample. And we can collect 3 to 4 leaves or petioles from each tree or vines. That means 100 leaves or petioles can be collected from 20 to 25 trees or vines for each sample. And these samples should be collected from 2 leaves or petioles that are located at the 4th or 5th position from the tip, basically from current year's growth, as seen in this picture. And as precautions, those samples should be kept separate for each cultivar, rootstock, and blocks of different ages. And obviously, we should not combine healthy and unhealthy leaves as a sample. Similarly, for strawberries, Late June sampling of fully expanded, recently mature leaflet is suggested for fruiting plants. However, for non-fruiting plants, leaflets are collected from early to mid-August. For blueberries, tissues can be sampled from late July to early August. And we collect mature mid-shoot leaves of current year's growth for blueberries. And for raspberries, we collect fully expanded leaf from fruiting cane in late July. And this table here shows sampling time as well as suggested plant parts for field-grown vegetables. This is also based on OMAFRA publication 611. For example, tomatoes can be sampled at early bloom stage. And at that time, we collect petiole from young but most recently mature leaf, which is usually the petiole of fourth leaf from tea. And then we can continue sampling every 7 to 14 days throughout the season. And for potatoes, samples can be taken 3 times in a season. Similarly, for carrots, petioles of young but mature leaf are sampled at mid-growth stage. For onions, tallest leaves are sampled minimum 3 times in a season. For broccoli and cauliflower, the midrib of young but mature leaf are sampled at the start of head formation. And for cabbage, the midrib of wrapper leaf is sampled at heading stage. And for celery, we usually sample the petiole of newest elongated leaf at the mid-growth stage. And for spinach, it is sampled at mid-growth stage, and we do collect the petiole of young but mature leaf as a sample. And for sugar beets, they are sampled when sugar beets are 12 weeks old, and we do collect younger but mature leaf as a sample for sugar beet. And regardless of these crops or sample parts, samples should be collected from at least 50 plants randomly from across the field, because labs need at least 250 gram fresh weight sample for processing and analysis. And sometimes we may require collecting samples for problem diagnostics. That means to determine or identify the problem and correct deficiency when needed. And in that case, samples are collected separately from normal growth and affected areas to compare healthy and affected areas. And care should be taken not to sample dead plants for diagnostic tissue sampling. And this slide is all about some do's or don'ts that we need to consider before, during, and after tissue sampling. And let me talk about some do's first. We should sample enough materials. That means 100 leaves or petiole for tree fruits and vines, and 250 gram fresh weight samples for field-grown crops and vegetables. Second, we should separate the petiole from the leaflet immediately to stop mobilization or translocation of nutrients after harvest. And third, we should put samples into paper bags, label them, and send to the lab for analysis. And some don'ts include not to collect chlorotic or dead tissue or tissues damaged by insect or disease. Second, do not collect plant tissues contaminated with soil, because contamination provides incorrect results. And third, do not ship the samples in plastic bags, because if we put samples into plastic bags, they will sweat and rot. So, some take-home messages from my presentation today are: symptoms can be confusing because many plant nutrient deficiencies share the same or very similar symptoms. Also, they can be similar to symptoms of many plant disease. Second, a certain way to know if a plant or crop is suffering from a nutrient deficiency is to have a soil test. Or we can also conduct tissue sample test for quick correction of deficiencies. Third, plant tissue analysis is most useful when it is combined with the soil test along with the visual inspection of the crop and soil conditions. And fourth, Tissue sampling methods differ with crop type, and therefore attention is required while sampling and handling of the tissue samples. So as I said earlier, this presentation is mainly based on the third edition of Omafra's Soil Fertility Handbook. And the PDF of this handbook is full color and available for free on Omafra website, while the hard copy is black and white and available for $20 plus tax and shipping from Service Ontario. Thank you for listening to my presentation today. So if you have any questions or comments, I would be happy to answer them. Also, you can reach out to me using this email. And at the end of my presentation, I would like to request all of you to visit soiltestmanager.ca, which is a new tool developed by OSCIA and Omafra for soil fertility recommendation. Thank you for your time again and have a wonderful day. Yeah, that's it. Unless there is any questions or comments, I hope the recording of the quality of the recording was reasonable today. And thank you very much for your time.
Yeah, thanks to Jendra. It was fine on my end. Looked great. There was just one question. What is the best way to determine if chlorosis is being caused by nutrient deficiencies rather than virus infection? And my chat just disappeared. And Katie, rather than viral infections, and then Katie responded, it can be very challenging. I would also always suggest taking samples and getting them tested for viruses known to infect the crop. And I didn't know if you wanted to add anything to Jendra for that.
I do agree with Katie. The most important thing is tissue sampling, right? So the visible symptoms are always confusing between deficiencies and disease. So it is always better to have tissue samples and have them sent to the lab for analysis.
Okay, great. Are there any other questions? I don't see any in the chat. So thanks to Jendra. And now we have Kristen, who will cover weeds.
Are you viewing my presentation view?
I see your presenter notes.
Thank you.
There you go.
Okay, hello everybody. Thanks for hanging in there. For my presentation today, you were supposed to go onto our YouTube Ontario Hort Crops channel and take a look at the presentation called Weed Scouting. And we're just going to do a short review of that presentation right now on some of the key items I think that will help you during your scouting adventure this year. And so let's just go through some of those questions. Okay, so Katie, can you put up the first poll question? Okay, the first question is, what do you call the first leaves that emerge from the soil? Are these called the first true leaves, broadleaves, cotyledons, or blades? Looks like we can end the polling now. Very good, those are the cotyledons. And here are some really nice pictures of cotyledons. If you can see that. The bottom one, the bottom picture here is Jimson weed. I think my computer may be frozen. Okay. There we go. And the top one is wild carrot. Okay. Okay, the second poll question is, what is the term for a plant that lives for 2 years? A summer annual, a winter annual, a biannual, or a perennial? Okay, looks like we can end the polling there. Like, good job again. 82% got biennial, which is right. Here's a nice picture of a biennial weed that is actually becoming much more common in all agricultural production systems within Ontario, and it's very difficult to control. This one is called wild carrot. So the next poll question is, I found a grass-like weed with a triangular stem. What is it? Is it large crabgrass? Is it quackgrass? Is it a sedge like yellow nutsedge? Or is it yellow foxtail? The key is triangular stem. Okay, looks like we can end the polling now. Good job again. The answer is yellow foxtail. So here is a picture of yellow foxtail in the field. You know, I don't know if you can really see the triangular stem, but yeah, that's what it looks like. Okay, our fourth question is, early emerging weeds compete more strongly with crops and cause greater yield losses than later emerging weeds. What is this important time called? Is it called the critical weed-free period, economic threshold, the pre-harvest interval, or the re-entry interval? Well, it looks like a lot of people did take the time to review the presentation, which makes me very happy. Okay, let's end the polling now. Great job. It is called the critical weed-free period. You are correct. And here you can see some of the differences in the crops with their critical weed-free period. But typically for horticulture crops, a critical weed-free period is for pretty much a really long period of time after the crop has been planted, because, you know, until they fill in those rows, and some crops don't fill in rows at all, like onions, you really got to keep those weeds under control. Okay, the fifth question is, what grass species does not have a ligule? Is it green foxtail? Is it Johnson grass? Is it barnyard grass? Or is it foxtail barley? This is a real key identifying feature for all grass species. You're gonna take that leaf blade back away from the stem to see if that ligule is there, if it's present, if it's membranous, if it's hairy, or if it's a mix of both. Okay, looks like we can stop the polling. The answer is barnyard grass. Great job. It is the only grass species without a ligule, so it is pretty easy to identify when it is small. And here we have that picture of barnyard grass with no ligule and the inflorescence or the seed head of it. Okay, question 6 is, what are the main identifying characteristics of broadleaf weeds? Are they A, cotyledons and first true leaves? B, leaf margins and leaf shapes? C, whether the leaves are alternate or opposite on the stem, or all of the above? Okay, great job, everybody. Looks like we can stop the polling. The answer is all of the above. And here, if you can recall from the presentation, we have all the different leaf shapes and how they are described, as well as a picture of what alternate or opposite leaves looks like. Okay, question number 7. What is the name of a new invasive weed that we should be made aware of? Is it purple pigweed, water hemp, purslane, or prostrate pigweed? Okay, looks like we can end the polling now. The answer here I was looking for was water hemp, but for those who answered purple pigweed, yes, this is also a new pigweed species that they found in Michigan. We have not found it here in Ontario yet. It is one to keep in the back of our mind when we are scouting if we come across something that's maybe a little bit different than red root or green pigweed. So yeah, that's not a, you know, for those who answered that, yeah, you are right as well. So here's some pictures of water hemp at about the stage where if you wait to spray to this point, you know, this is when you need to spray right now. Don't let this plant get any bigger. And this is me actually standing in a field, and that is a soybean field. Can't barely see the soybeans, and it's just full of water hemp. In Essex County, where we have about 37 fields, I think, confirmed now with water hemp populations. Okay, question number 8. What weed species can cause blistering? Wild parsnip, wild carrot, garlic mustard, and lamb's quarters. Okay, looks like we can stop the polling. Yes, the answer is wild parsnip. Wild parsnip is in a relation to wild carrot. It is also a biannual, meaning it overwinters as a rosette and then the next year produces flowers and seed. If you even just brush up a little bit against this plant, if you're walking, you can get what's called photodermatitis, which is like get a little bit of the resin from the plant on your skin, and then you go in the sunlight, you can get a rash. And just like with poison ivy or any other irritations from plants, everybody reacts a little bit differently in terms of how serious that rash can be. With this one, you can get blisters and bubbling. It can be quite sore. It has happened to me. Just that little piece of skin between my sock and my crop pants that I was wearing one day, and I was walking, taking pictures of wild parsnip. That skin right around between my sock and my crop pants, I had a rash just in that one spot. So, you know, you just need to be careful. Mine wasn't very serious, luckily enough, but there has been cases where it can be quite serious. And this weed species has been thought to, for the longest time, thought to be a weed problem in eastern Ontario, but it has moved down all the way to the southwestern part of the province, lots in Essex County now, Chatham, Kent, because it moved along the rail lines. So it was transported by trains. And now we actually have it a lot in orchards, which becomes a issue for labor and your workers in the orchard. So you need to be really careful with it and make sure you get it under control. So that's it for the review of scouting for weeds and some key identifying features of weeds to help you in your scouting this year. I'm always available for questions and can be reached in a number of different ways, by email, by text, or my cell phone there, or at Twitter. So you can send me pictures of things you can't identify, but please use the tips that you learned today when taking pictures, because it's very frustrating a lot of times when you get a picture and you're just like, I think it's this, but I just don't know because I cannot quite see a clear picture. So with that review, if there's any questions, I'll take them now before I head into the next presentation.
I don't see any questions yet, Kristen.
Okay. Everybody did a great job on those questions. It did a great job in review, so good job. But there's so many different weeds and so much to learn. I know it can be a bit overwhelming, but you know, all you do is if you don't know, you ask questions, you write it down, and you make sure that you report it to your supervisor, who can then make recommendations to the growers. Okay, So now I'm going to talk about the importance of scouting for weeds after herbicide application. It's always important to scout on a regular basis. You've heard this all day long today. And
Sorry, Chris, don't mean to interrupt, but it's just we're seeing your presenter view, so I don't know if you want to swap.
Same settings. It's funny how that happens. Okay. Thanks, Katie. So, yeah. Oh, where was I? Okay. It's just really important to be consistent in your scouting and to do it on a basically a weekly basis if possible, to get to those fields to see changes, because things change very, very quickly over the summer. Okay, so these, I should also mention that these slides have come from the Weed Science Society of America training and extension page. So you can go there to their website and you can find a lot of really good, valuable resources about weeds and scouting and lots of fact sheets. So if you're really interested in weeds like me, it's a great resource for you. So there are many, many factors that can contribute to the presence of weeds in the field after herbicide application and later on in the growing season. But scouting is the only way to know which weeds are present, and their patterns in the field can help you understand why they are present. Scouting will be helpful in documenting changes in the weed population that occur over time in response to land management practices, including the evolution of herbicide-resistant weeds. Changes in crop and weed management practices based on scouting help to maximize crop yield and profitability by reducing weed competition, reduces weed seed production, and maximizes remedial management tactics within the same growing season, which means, you know, basically within that growing season, you'll be able to do something about those weeds that exist in the field right away, so you're not perpetuating the problem year over year. So when you're scouting for weeds, you should scout 7 to 14 days after each herbicide application and continue on regular intervals until harvest. You should move across the field in a scouting pattern covering the area. It could be a zigzag pattern, a Z pattern, a W pattern, but you really should make sure that you're hitting all areas of the field, from the field borders or edges, the entry point of the field, as well as the middle and other parts of the field. You should always target low and high areas within the field as well. So what you need to do, you need to basically record what weed species is there. If you do not know what weed species is there, record whether it's a broadleaf or grass. Make sure you record what growth stage it is in, whether it's, you know, cotyledon, first 3 leaves, 2 to 4 leaf, or if you can't really tell, because sometimes with grasses, the tillering makes it difficult to know like how many leaves it is, measure it. Make sure you take the height, because a lot of herbicide labels for the growers will say must be sprayed before 10 centimeters or 15 centimeters high, and they don't typically go by leaf stages a lot of times. You should also record the spatial patterns of the weeds across the field. So are they in clumps? Is it just an individual plant here and there? As well as the number of weeds in a specific area. And also whether there's live and dead weeds after the herbicide application. So if a whole bunch of pigweeds are there and half of them are alive and half of them are dead, something is going on. And then you should also write down what the herbicide symptomology on those weeds are. So is there any chlorosis? Is there no symptomology? It looks totally healthy, or is it like on its way to die? So there's a lot of information there that you need to write down. And I have included in the handouts a nice checklist for you to go through when you're scouting for weeds in the field. So if weeds are present after an application, you need to really determine the reason. Okay, you need to figure out the field history. Has this grower been doing a monoculture, like just growing the same crop year after year after year? Or is it a short rotation? Is it tomatoes, rye, or potatoes, rye? Or does he have like a long diversified crop rotation, including 3 or more crops? You need to think about the weed biology. Is this an annual? Is it a perennial? Is it a biannual? You need to think about the environment. What was it like when the grower sprayed that field? Did enough time pass before it rained? Or was there enough rain after he applied the product to make sure it was incorporated in the soil? Depends. Was there any application problems? Is there a section in the field where there's just a whole bunch of weeds? Could have been a plugged nozzle or something else. Could have been a bump in the field or a rock that the sprayer ran over and missed a section. What are the crop cultural practices? Is it no-till, or are they tilling? Or the very last thing you should think about is herbicide resistance. Is there a history of herbicide resistance in this field? Is there a history of the grower using the same herbicide groups over and over? Or if they do grow the crops in a very short rotation, then that grower is continually applying the same herbicide groups year after year. So even though the crop is different, the weeds are the same. And so they're getting that same, they're getting the same herbicides over and over, so you're not mixing it up. And so basically you need to think about the whole system, everything that's going on before we, suspect herbicide resistance in the field. So here's a checklist, an abbreviated one for weed scouting. Scout 7 to 14 days after each herbicide application and near harvest. Identify and record the weed species that are present. Determine the distribution pattern of plants in the field. Correlate with application pattern, identify as random. So what that means is basically, say for example, there was a plug nozzle. Well, then you would probably see a strip going down the field one way, and you'd probably see a strip of weeds coming back the other way, just because that one nozzle was plugged and there was no coverage over those weeds. If there's a random pattern, then it's probably something different. It doesn't have to deal with the equipment. Determine if the plants present survived a previously applied herbicide or emerged after the last herbicide application. So this is a little bit tricky, and is definitely harder if you're not visiting the field within 7 days usually, especially in the middle of the summer, because everything is growing extremely fast and weeds can grow extremely fast. Like stuff like water hemp can grow like 2 to 5 centimeters in a day. So, if you're not there really often, it's hard to tell whether or not, those weeds were there when the grower sprayed or just emerged afterwards. So you need to make notes about that. And like I said earlier, observe individual plant responses, especially if plants survive the herbicide application. So if some plants survive that would normally have been controlled, you should look very closely across the fields for other plants of the same species to determine, if there's a varied tolerance response to the herbicide. And then you need to look at the previous field history to understand why or what's happening in the field, changes that might be occurring. So here's an example of this, in terms of, there's some variability in the tolerance of the weed to the herbicide. And so you got a varied response here, you can see. There's no symptomology. You can see the stunting, and then you could see plants that are dying. So in a field, when you come in like this and you see a huge patch of weeds right there, that's something that you definitely would want to go in and investigate. So in conclusion, scouting fields to determine the reasons for weed survival after herbicide application is really important. You need to know the field history, the weed biology, environment, application parameters, the crop culture practices, and herbicide resistance are all factors that can contribute to weeds surviving the application of a herbicide. Symptomology may differ between the observations of low-level and high-level herbicide resistance, and that was what I was showing in the previous picture. Confirming herbicide resistance early when just a few weeds are present and removing them by hand can decrease the spread of herbicide-resistant weeds, thereby reducing the cost required to manage them. So this is really important because, as we know, herbicide-resistant weeds are not going away. They are only becoming more and more problematic. And with challenges with reevaluations and the limited amount of post-emergent herbicide options that horticulture growers are dealing with, it's a very serious issue that we have. And in Ontario right now, we have 21 weed species that have some level of resistance that's been confirmed across the province. The scariest trend is that a lot of weed species, particularly those in the Amaranthus genus or the pigweed species group, there are many fields in Ontario with multiple resistance or resistance to 2 or more herbicide groups. So, these are things to be cognizant of and be aware of, because horticulture crops, even though we rotate crops, we have very few herbicide groups. It's around 7 that are pretty common, that are used frequently in horticulture crops that, we're rotating crops, but a lot of times we're not really rotating herbicide groups. So resistance is a problem, big problem. So because of that, we've been working on creating genetic quick tests for herbicide resistant weeds. And if you ever do suspect that you have this problem, or you should talk with your supervisor and then have them contact me to get some sample collection kits so that we can get those weeds tested quickly. Usually this takes anywhere from 2 days to 2 weeks. Usually we hope it only takes like less than 7 days, so that you can make a management decision in the season, and hopefully the grower can do something different to control those resistant weeds and not just have to hand weed them out. So I strongly encourage you to, talk with your supervisor and then contact me if you want to get some testing done. It's really easy what you need to do to collect the samples. We only take like quarter size leaf tissue from 10 different plants in the field. So you'd want to be very random in your collection of these leaves. And then each leaf tissue is put in an envelope with a label on it that I want you to fill in some information there. And then we put it into a Ziploc bag with silica gel. And the silica gel is just like a desiccant. It kind of takes out the moisture from the leaf tissue, so they don't mold or anything by the time they get to the lab. But because the lab is in Ontario now that we're using, it gets sent there really quickly. And the lab that we use, if you're interested in all the different tests and stuff that they do, they don't just do weed genetic testing. There's a lot of different genetic tests they do, including the Rugose virus test that Katie talked about. There's their website. And I have project funding for this for at least this year again. So I strongly encourage, if you suspect there's resistance in a field, that the grower get those plants tested. It's easier to deal with something that you know what you're dealing with than just kind of guessing that maybe that's what the problem is. Because if you leave this problem for too long, basically the seed bank just builds up, and then the grower will be dealing with herbicide resistant weeds forever, basically. So it's a really good idea to get it tested, know what you're dealing with, and then move on. And I also encourage growers not just to take samples from one field on their farm. I think they need to get a resistance profile for every field on their farm, because with things like water hemp that can be resistant to four different herbicide groups, you might have a field where there's not that. There might be one with resistance to 4, one with resistance to 2. So it's free, and a lot of people say free is for me, so why not get the information? Because knowledge is power. Get them tested so you know what you're dealing with, so the grower's not throwing money out the window by applying an ineffective herbicide. So there's definitely a lot of weeds in fields across Ontario, so you're going to really need to learn how to identify them. And this is an excellent book put together by my colleague Mike Cobra, who's a field crop weed management specialist, and Dr. Francois Tardif from the University of Guelph, and his wife Jocelyn Latart. And you can order this book by Googling it. Go to Weed ID Guide for Ontario Crops, and it'll take you to a website called Blurb, and it's about $37.50 with tax and stuff. But if you order more than 10 copies, then you get a discount. So that's pretty cool. And of course, also there's the Guide to Weed Control for Horticulture Crops 2021. There's a free PDF download available, or you can go to the Publications Ontario and order that as well. And so with that, I'm here as a resource for you. Send me questions, like I said, by email, by text, by Twitter. You can direct message me, whatever you feel most comfortable with. Take those, recommendations about how to take good pictures. That always helps. I get asked a lot if there's a good weed ID app where you can take a picture and then it tells you what it is. And the quick answer is no, we're not there yet in terms of that. We've done some testing, my colleague and myself, and we know what the weed is. We take the picture and it comes back as something different. So, there's more work to be done. We'll get there eventually, but it's not quite there yet. So use the knowledge base you have. Don't be afraid to ask questions. There's never a dumb question. So ask away. We're here to help. Thank you.
Great, thanks, Kristen. Are there any questions? I don't see any in the chat yet. Okay.
Very quiet group today.
Yeah.
Okay, well, ask any questions later when you think about it. No problem at all.
Okay, well, thanks, Kristen. And the last short presentation will be by Katie.
Okay, congrats. Yeah, sorry, congrats, you made it to the end. We're just gonna quickly go over some handouts that were sent out in that package, so you have them for reference. Just gonna refer to them here. As Kristen mentioned, you got a lot of information today, but we're all here to help the growers. So we're references. If you don't know what's going on, take good pictures, get samples, reach out for help, because no one expects you to know everything. So we're here to help with the grower managing some of these pests and issues. So you have these in your handouts. I just want to quickly go over this first poster, which is common plant disease symptoms. So we went over this morning an intro on plant diseases and plant pathogens. There's some common terms here that we use when we're describing symptoms. So necrosis is brown dead tissue, chlorosis, yellowing, water-soaked equals dark wet lesions. There are pustules, which are really common in the rusts that we discussed this morning. There are spots or lesions that are really defined, which are really common symptoms of plant diseases. There are wilts, which is typically from a soil-borne organism that's causing an issue in the root system, and that causes the above ground plants to wilt. There's blight, so that just refers to the rapid killing and decomposing of leaf tissue. There's rots, which typically refers to the fruit as well as roots. There can be root rots as well, so succulent roots and fruits. That's a very common disease symptom. Galls and knots are also can be quite common with some of the diseases that produce overproduction of some sort of hormone, which can result in the swelling of that tissue. There's discoloration, which is common in a lot of our viral infections and some other pests. There's damping off, which is referred to as pre or post-emergence rotting of the seedling. So this is what we refer to really early in the season. So weather like this, if there's crops that are out there that have already been planted, this is really, this weather really promotes damping off. So the cool, wet conditions that allow soil-borne organisms to rot out those roots and kills that entire plant early in the season. And then cankers is another common symptom of plant diseases. So you can see here, this is a fire blight canker, and what probably happened was this shoot was infected and it led all the way down to the main trunk. And so you can look for those on those perennial crops. And then there's one here, you can see cankers on the stem there of that succulent tissue. Okay, so The next handout that you have that I just quickly want to touch on is scouting in plant diseases. Let me get this in full screen. So you've heard a lot about scouting for other pests. It can be quite similar when you're looking for diseases, just you're kind of looking at an overall range of what's going on the farm. So again, similar to other pests, why you want to scout for diseases is for timely management for that grower and limiting the spread of some potentially really impactful pests. So scouting tips, like other ways of scouting, right? This is kind of an all across the board. You want to know the weather events. You know, the environment really can help figure out what kind of disease is present or when it's showing up. Look for hot spots. So when a disease or an organism is causing the issue, it's typically in patches or in spots of the field as opposed to across the entire crop. And so it's really important that if you see a hot spot to investigate that. But you also want to make sure that you're looking in that field in a random pattern, so a W, an X, a Z, to look if you're not seeing any, you know, distinct spots or distinct issues, that you're walking through that field in a random fashion and taking samples. So you're checking frequently for plants, frequently for issues. Make sure you're getting down in there, you're looking on the top of the leaf and the bottom of the leaf. You know, this can be important for diseases, but also insects. A lot of them hide out in the heat of the day underneath that leaf in the cooler canopy, so really important to look under there. You know, check all the different limbs. If you're working with a really big crop, like a tree fruit, make sure you're looking up and you're looking down. And there's some really good tips in some of the individual sessions and videos for those kinds of crops, because, you know, they talk about it when you're going into the crop to look at it as a whole picture before you get so specific, because you can get kind of blinded if you're not looking at everything before you walk in and look for something very specific. Where possible, so for some of the field annual vegetables and crops, you can pull out a plant and look for signs of root rot, so discoloration of the roots or the crown in some crops where they have crowns. So if you have a knife, make sure that you're cutting through the roots and stems. And as my dad says, always cut towards your friends, not to you. Make sure you're careful when you're using that knife. But you're looking for signs of discoloration that could lead to wilting and rotting. Always keep good records, just a general tip, and always make sure you're communicating regularly. Denise mentioned a lot on biosecurity, really important for a lot of the soil-borne organisms, especially on your boots. I carry tons and tons of boots around with me if I'm going to multiple farms, just in case you don't have time to clean those. But I think that's really, really important, especially for some of those soil-borne diseases. And then collecting samples. So when you're collecting samples for a disease, you want to collect plants with the symptoms. You want to place them in a bag and label it correctly as to where you found it. The key here is for some of these diseases that have spores, they could be actively sporulating. You don't want to transport those disease samples around the field where possible, because you could be moving those spores unintentionally throughout the field. So carry bags with you, put that sample in a bag, and then continue to walk around the field. And then keep these plant tissue samples cool, not frozen, but cool and out of sunlight so that they don't rot completely before they get to the lab. And it's really important to get them to a diagnostic lab fairly quickly. You also don't want fully dead or almost completely rotten issues, because there's often secondary things that get in there. So you want something that's showing symptoms but still has some healthy tissue with it. So that's really important when it comes to disease and for shipping samples. So I went through that pretty quickly. Always here for any questions or for any assistance, but I will stop sharing now and take any questions. No questions. All right, that's fine. Just feel free to email or text anytime.
Okay, yeah, I don't see any questions. Thanks everyone for participating in the workshop today and hanging on to the bitter end. And thanks to my colleagues and Elizabeth for sharing their knowledge today. And I hope you enjoyed the workshop. I hope you learned a lot. If you do have any further questions for us, we mentioned quite a few times, feel free to reach out to us. I did include our contact information for all of us in the handout that I provided to you, that Adobe link that I provided. So hopefully everyone could access the documents there. I haven't heard any